Recording now, if anyone wants to watch it later. So I have some general information here on the test, in case you don't know. So it's gonna be April 6th from 5 to 7. Bring your pencil, student ID, and it's gonna be in Lee Hall 107. This is what she told me the breakdown is. I don't know if she changed it, if it's changed by now, but as far as I know, it's gonna be 10 questions where you match the enzyme with the pathway. These ones should be pretty simple. It should be just memorization. And a lot of them are very uh, self-explanatory. Like, for example, if you have like pyruvate kinase, you know the enzyme has to do something with pyruvate, so you're able to know what pathway it's in. So I think this will be the easiest part of the exam. And then you have 33 multiple choice. I'm not sure what the difficulty or how she will structure this, but she said they will come all from the problem sets. So that's what we'll go over today, and we'll try to cover that section. There'll be 4 multi-select, and I think the way she said that will work is she'll give you a scenario, and one example she talked about, for example, is if insulin is released, you would be given a certain number of choices, and you would have to say if they would occur or they won't occur. So you would have to have more understanding to answer this one, but hopefully after going over what we do today and then going over the stuff I posted, it'll help. And then 5 open response, that's probably going to be the hardest part of the test. You have to know your stuff for that one. The worst part about this is she can pick any of the questions from the problem sets. So it forces you guys to have to pretty much know pretty much everything to answer this part of the test. When I did the problem sets, I included some things that I think she might ask about the open response. Okay, so in this, the way I structured this is I just have the problem sets, and I have we'll go over them, explain them, and then I'll ask if anyone has questions on the problem set, and then I have some questions you would potentially ask. Okay, so that's how I structured this. So this is problem set 1. This is question 1. If you want to like follow along and pull it up, this is from her answer key. So the first question is talking about metabolic concepts, and she wants an example, either in glycolysis or TCA. The first one she has is concept of changing location. So there's a lot of examples on this. The one she gave is oxaloacetate. If it's in the mitochondria, you're going to be running TCA. So that's normal TCA stuff. Oxaloacetate, and then comes in with acetyl-CoA, and then you get citrate, and that's the first step in TCA. And then if you have oxaloacetate in cytosol, that's your gluconeogenesis stuff. So that would be you would first convert pyruvate to oxaloacetate through pyruvate carboxylase, and then you would do PepCK to get oxaloacetate to make PEP, and then you would run gluconeogenesis. And then you do that over past the enzyme pyruvate kinase. So I think for this question, she's just asking about the change in metabolite. So that would be the first example. Another example I can think of as well is, it's not about glycolysis and TCA, but remember fatty acid synthesis occurs in cytosol, and then beta oxidation is in the matrix. Okay? In matrix. So that would be a second example I can think of. So that way, beta oxidation, fatty acid synthesis, because they share a lot of the enzymes and similar pathway. They're regulated based because they're one way to regulate them is to put them in different organelles. And then another one I can think of is citrate. Very similar to oxaloacetate. If citrate is in the mitochondria, you will run your normal TCA, and if citrate is in the cytosol, you will be running fatty acid synthesis. So remember, citrate goes into the cytosol through the tricarboxylase shuttle when you want to run fatty acid synthesis. So that's another way, change in metabolite location. I think the other one I had, can't remember, but I think the other one I had was beta oxidation. So this is not specifically change of organelle. This is change of organ. So beta oxidation occurs in, or not beta oxidation, ketone body synthesis occurs in the liver, and then ketone body breakdown occurs in other tissues like the brain or muscle cells. So that one is not change of organelle, but it's a change of organs, but it's still considered a change of location in the body. Okay, so part B is reaction. So she wants a concept reactions far from equilibrium that confer directionally on a metabolic pathway. And she has hexokinase has a large negative delta G. I think what she's trying to test in this, these sound really familiar now that you guys are almost like halfway through the semester. But just be able to identify enzymes that have negative delta G. So a question she might ask is she might give you which of the following enzymes, and you have to say which one has a negative delta G. So that would be an example she can ask. So citrate synthase is negative in TCA, and then isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase. And remember, you need to know the three ones in glycolysis as well. So you have pyruvate kinase, hexokinase, and then PFK. Okay. 1C, so reactions that can be coupled to make a non-spontaneous process spontaneous. So know about the coupling. So citrate synthase is coupled in TCA, and then any reaction that uses ATP as well, that usually couples two reactions together. And then the last concept she wants to know is a substrate cycle that allows fine-tuning of metabolic flux. So this is, so if you remember in glycolysis, you have PFK and fructose 1,6-bisphosphate. So make sure you know this substrate cycle and how that prevents futile cycles, depending on what the cell needs. And then there's also the glycerol 3-phosphate shuttle. This one, so that's the one where it allows you to make NAD. Let me see if I can pull it up really fast. It's this one. So remember glycerol 3-phosphate, the cytosolic form, and then glycerol 3-phosphate dehydrogenase, the mitochondrial form. So those ones can also fine-tune our between DAP and glycerol 3-phosphate and between NAD and NADH. Okay, and then the last thing is urea cycle. So urea cycle, we also talked about a transaminase, and then we have glutamine synthase. So those two reactions can regulate between the levels of alpha-ketoglutarate available in the cell versus glutamate that is available. Okay, you guys have any questions on this one? All right. I think I put too much detail into this. I might have to move a little faster if we want to go over every question that there is. So the next question she has, pyruvate going to lactate. Draw the structures of pyruvate lactate, and she wants you to know if it's a oxidation reduction reaction, and she also wants you to explain your answer. Okay. So I also listed the pathways for all of these problems, so it's easier for you when you're going back to study. The pathway this is in is glycolysis. Remember pyruvate. When you're running anaerobically, goes to lactate, and this is done by lactate dehydrogenase. Okay. So a question she might ask you based on this, she could draw the structures for the following reaction. So she can give you pretty much any reaction in TCA or glycolysis, and you would have to be able to tell what substrates the reaction uses. You also have to know what kind of reaction it catalyzes, because this reaction asked if it's oxidation or reduction. So to figure that out, it's really, I kind of give you this summary here based on the name of the enzyme. So if the enzyme has dehydrogenase in it, it will do oxidation reduction. If it has a phosphatase, it will remove a phosphate. If it's a kinase, it adds a phosphate. Carboxylase adds CO2. Decarboxylase removes CO2. Transaminase removes and adds NH3. A mutase is an isomerase, so it takes the same molecule and switches, transfers groups around in it. Synthase, it's in its name, it synthesizes stuff, so like glycogen synthase. Phosphorylase uses free phosphates to break down stuff. So an example that would be like glycogen phosphorylase. You also have hydrolase. Those break down stuff using water. So if you have no idea what the enzyme is or you've never seen it before, you can use these to help you have an idea of what the enzyme could potentially do. Another way she could ask this question is she can give you a reaction and you would have to say what the enzyme is. So you have to know the substrates and you have to know what the enzyme does. So that's why I said it's really important to know all the enzymes in glycolysis and TCA. If there's enzymes and substrates you should study, it should definitely be the ones in TCA and glycolysis, because personally I think these are the most important ones and are mainly going to be the ones where she asks questions about this stuff from. Okay. If you guys have any questions, feel free to interrupt whenever you want. Okay, question. Oh, wait. Go ahead, Angel.
Do you think we should, when you say like know those enzymes and substrates, you mean like not only the names and stuff, but like also the structure?
Yeah, we had to know the structures for our test. So I
Okay.
think you have to, for structures, know glycolysis and TCA for sure.
Okay.
And then for everything else, know substrate names and then enzyme names. Okay, question 3. So this is reduction potential. It gives you a mixture of malate, oxaloacetate, NAD, NADH, and it wants you to know which compounds oxidized or reduced. Okay, and you have to explain your answer. So she said oxaloacetate will be reduced to malate, while NADH will get oxidized to NAD. Therefore, electrons will be transferred from NADH to oxaloacetate, and the reduction of oxaloacetate has a. And then she explains why this happens. So this pathway is in TCA. I think this question might be asked mainly because it can be really tricky if you don't know what she's talking about. So whenever malate goes to oxaloacetate in TCA, this reaction generally has a really positive delta G. Positive delta G. But the only reason it runs in this direction is because it's coupled with other things in the cell like citrate synthase. But if this reaction is left by itself, if you isolate this reaction, it will actually favor the other direction because it's positive from malate to oxaloacetate. So by itself, it will favor the malate direction. So that's what she's trying to test you when she's asking this question. And then another part of that question is you have to know what's being oxidized and being reduced. So the best way to do these is a lot of times these will use cofactors like NAD, FAD, or NADH or FADH. An easy way to remember this is NAD is the oxidized form and NADH is the reduced form. And same with FAD and FADH too. Okay? So if a reaction has like a substrate, let's say we have NAD plus X, you have no idea what X is. It's going to like a different substrates and NADH. Because you know that NAD is the oxidized form, so this is the oxidized, and you know NADH is the reduced, you can easily tell what these substrates are without knowing their structure or even their names. So you know that the Y is going to be the oxidized form, and then X is going to be the reduced form. So I think that's the easiest way to identify a reaction and be able to tell what's being oxidized and reduced is if we just follow NAD and NADH, we'll be able to tell what the other substrates are. So for example, in this example, this would be oxidized to this product because this is the oxidized form. Okay? And another easy way to remember it is by looking at alcohols and ketones. So if you know the structures, for example, the reaction between pyruvate and lactate. Pyruvate gets reduced to lactate in that reaction. And pyruvate has the ketone and lactate has the alcohol. So whichever one has the alcohol is the reduced form, and whichever one has the ketone is the oxidized form. So that's one way to remember these reactions to help you answer if she asks what's being oxidized and what's being reduced. Okay. Question 3. For each of the following enzymes, draw the structures, reactions, and products in cofactors. All that stuff. And then she also wants you to describe the thermodynamics of the reaction. Okay, so because she can ask any question like this, where she gives you just the enzyme name and you have to say, does it say draw the structure? Yes, it also says, yeah, draw the structures of the reactants. So for these 4 enzymes, for sure, you have to know their structures and their delta G in case that shows up on the test. She could also ask, so instead of like malate dehydrogenase, she could ask like fumarase, for example. So that's why I think it's important to know all the structures in TCA and glycolysis, and you also have to know their delta Gs. But if you're low on time, you should study the ones she puts in the problem set. Okay, so briefly, malate dehydrogenase takes malate, converts it into oxaloacetate. That's the one we talked about in the last example. Glucose 6-phosphatase, so that's, remember, the opposite of hexokinase that takes glucose 6-phosphate and converts into glucose. Citrate synthase, remember that one takes oxaloacetate and acetyl-CoA and makes it into citrate. That's the first enzyme in TCA. And then succinyl-CoA synthase, so this is one in the middle of TCA. It's at the bottom, like right here. So if this is TCA, it's right here. That one takes succinyl-CoA and converts it into succinate. Okay? So I would know all the enzymes in TCA and glycolysis in case something shows up that you weren't expecting. So I have possible question: the following enzyme catalyzes which reaction, and then draw the following structures for the following enzyme as a possible question. Okay. I think so far it's pretty self-explanatory. So next she gives you the delta G of citrate synthase, and you have to calculate the equilibrium constant, and she gives you the temperature too. So you have to be able to use the equation. I think this delta G is equal to negative RT natural log of K. So if you're given delta G and temperature, usually R is 8.314, you have to be able to convert that to equilibrium constant. Also, make sure you also know how to do it the other way as well. So if you're given equilibrium constant and temperature, make sure you can convert to delta G. Okay? Would you expect this reaction to be a control point in TCA for part B? For control points, usually they're going to be enzymes with negative delta G. So if you don't remember the ones in TCA and glycolysis, if the delta G of it is negative, it's going to be a control point most of the time. And if it's around 0, most of the time it's not going to be a control point. And if you do want to remember the enzymes in TCA and glycolysis, the ones in TCA that are regulated are citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. And in glycolysis, the ones that are regulated are glucokinase, PFK, and pyruvate kinase. Okay? And then negative delta G enzymes are usually regulated. And then make sure if you're given delta G and temperature, you're able to use the Nernst equation. Actually, it's not the Nernst equation. Nernst equation is a different type of equation to calculate the equilibrium constant, or the other way around. So that's for question 4. Okay. Next question. Oh, go ahead, Ellie. Yep.
For the delta G's, are these the only ones that are like negative enough that they're like spontaneous, and the rest of them are just like around 0?
For glycolysis in TCA, yes.
Okay, I just want to make sure.
Yep. These are the ones that are regulated and usually are negative enough that they're relevant.
Okay, thank you.
Mm-hmm. Also, I will point out, if this is given in kilojoules and use 8.314, 8.314 is usually in joules, so make sure you convert the kilojoules to joules. Just something to keep in mind when doing this. So make sure all your units match up. Okay, so question 5. She gives you the standard free energy for malate dehydrogenase. This time it's really positive, so it's non-spontaneous. And then she asks, would this reaction occur spontaneously in the cell? It probably won't because it's really positive. So yep, she says because the delta G is greater than 0, the reaction won't happen. And then she also wants you to explain how citrate synthase promotes the malate dehydrogenase reaction. So you have to understand the idea of coupling for 5B. Usually in in your body, when you have an enzyme with a positive delta G, you usually couple it with another enzyme that has a really negative delta G. So the overall free energy change is negative. And I think that's what she wants in part C. She's asking what is the overall change in free energy for the two reactions. This is malate, this is citrate synthase, and then you add them and you get negative 1.8. So overall it's negative, so they will run in this direction, and that's why TCA runs in that direction. So this is in TCA, positive delta G is non-spontaneous. A question I have is she could ask which of the following reactions is spontaneous or non-spontaneous. So you would have to know the delta G's for these types of questions. She could ask what enzyme is malate dehydrogenase coupled with. So that would be citrate synthase, and she would give you different options of enzymes. Malate dehydrogenase has a positive delta G. Explain why it runs in unfavorable direction. So you would have to talk about. So yeah, malate dehydrogenase has a positive delta G, but it favors the unfavorable direction when it runs in the cell. So you would have to talk about why that is. So you would have to talk about why citrate synthase. When citrate synthase couples it runs in that direction. So that's all you would have to say for this kind of question. Malate dehydrogenase has a free energy of. So when I took this class, we had to know delta G's of certain enzymes. So you had to memorize them. You can just say they're negative or positive. So I don't know if she would ask for that, but if she ever asks for malate dehydrogenase has a free energy of, you can just say positive. And citrate synthase has a negative delta G, you can just say negative. But they're negative 31 and positive around 29 for malate dehydrogenase. Okay. For question 6, I think this is hypothetical. She gives you a bunch of metabolites, and she tells you they're catalyzed by these enzymes respectively. So you have A, B, C, and D. This is catalyzed by X, this is catalyzed by Y, and this is catalyzed by Z. And you just have to answer questions on them based on your knowledge of biochemistry. So you have to know the idea of coupling and the delta G's. So for the example she gave us, this one is around 0. This one is really negative, and this one's around 0. So Y is going to help. So enzyme Y will help couple the other enzymes in this direction. So that's what I mean by the idea of coupling. And then for part 6A, she's asking which one is the main regulatory enzyme. To answer that question, you have to know that when an enzyme has a negative delta G, it's the regulatory enzyme for that pathway. A question she could ask from this problem set is which of the following enzyme is the regulatory enzyme, and she might give you a couple ones in TCA or glycolysis. Or she could give you a random pathway with delta G's, and you have to know which one is the regulatory one. So if she does that, the enzyme with the negative delta G would be the regulatory enzyme. And then for TCA, there are 3. Remember, citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. For glycolysis, you have 3 that are regulated, but you have 1 committed step. So your committed is PFK, and your other ones that could be regulated are glucokinase or hexokinase and pyruvate kinase. And then, so for 6B, she asked if an enzyme is inhibited, how would that change the concentration of the different metabolites? So if you're given a scenario, random one, you would talk about the concentrations of the different metabolites and how they would change. So for the example she gave you, she said if we add an inhibitor to enzyme Z, so this is enzyme Z, how would that affect metabolites A, B, C, and D? Okay. So at this point, you should be able to answer this question pretty well, but I'll go over it briefly. So if Z is inhibited, this won't affect A, and it won't affect B. It will mainly affect the two metabolites around it. If Z is inhibited, you're not converting C to D, so D will decrease and C would increase. So that's all she wanted for this question. Okay. 7. This is one of the questions I missed when we took our test, by the way. But I think I understand how to do it now. So for 7, it says pyruvate dehydrogenase complex is the critical enzyme for converting. So pyruvate dehydrogenase complex, remember, converts pyruvate into acetyl-CoA. So this is right before you do TCA. So this enzyme kind of links TCA and glycolysis. Glycolysis is up here, and then TCA starts down here. Okay. So that's where the enzyme is, in case you can't remember. So for this one, so describe the set of reactions that occur at E1, E2, and E3. And so basically for this, you have to know what E1, E2, and E3 does. So I think this could be one of the potential open responses, because pyruvate dehydrogenase complex, they put a lot of emphasis on it. So I would know this for sure. Briefly, E1 catalyzes the removal of CO2. And remember, you make the TPP intermediate. You have acetyl-CoA. Actually, let's see if I can pull it up real quick. Doesn't take too much time. Right here. Okay. I like this more. So we have pyruvate. We lose a CO2. We make a hydroxy TPP intermediate in E1. Okay. After that, we have. Sorry, I lost my train of thought. We have pyruvate. We lose CO2, and then we make hydroxy TPP by E1. And then E2 takes that, uses the lipoamide cofactor, and we make another intermediate called acetyl dihydrolipoamide. In that process, we send TPP back into E1. And then E2 does another set of reaction. It takes CoA, it takes acetyl dihydrolipoamide. We make this dihydrolipoamide, and then we release acetyl-CoA. So this is one of the products. And then we need to regenerate this dihydrolipoamide back into a lipoamide. So E3 does that. And in that process, E3 gets reduced when it does that. And then it uses NAD to NADH to get oxidized back into the oxidized form. Okay. So that's just a brief summary over it. I have a video on it if you're still confused on that. But I would know the whole process of pyruvate dehydrogenase complex. Indicate how this enzyme is regulated by post-translational modification. So this enzyme can be, do I have it? No, I didn't add it. E1 can be phosphorylated by pyruvate dehydrogenase kinase. And then here, this is better. So pyruvate dehydrogenase complex can be activated by a phosphatase, and it can be inactivated by a kinase. And there's other things that can act on the kinase too, and the phosphatase. But just know when it has a phosphate, it's in the inactive form, and when it's dephosphorylated, it's in the active form. Okay, so that's how it would be regulated. And then a researcher says that pyruvate dehydrogenase complex could be a source of acetate, and based on your answers to the first part of the question, why would that be highly unlikely? So this would be highly unlikely because we would have to break the thioester bond that we made. I think that was my explanation for it. I think she said acetate is never a product because acetyl group is attached to the lipoamide cofactor and then transferred directly to the other sulfide group. Mainly because if acetate is the product, we would have to break the high energy thioester bond that we make in pyruvate dehydrogenase complex. So it would negate the whole point of the pyruvate dehydrogenase complex if acetate is released. Okay, so for possible questions you could literally ask anything about this. She could draw in full detail the steps of pyruvate dehydrogenase complex, which I think she might do. She could ask which complex uses what cofactors. So she could ask which complex uses TPP. So for that, you have to know your cofactors. E1 uses TPP, E2 uses lipoamide, and E3 uses FAD. 7b, she could ask under low energy conditions or high energy conditions, would pyruvate dehydrogenase complex be active or inactive? So under low energy conditions, you would want to run pyruvate dehydrogenase complex to enter into TCA and make energy. So in that case, pyruvate dehydrogenase complex would be in the active form, which is the dephosphorylated form. And then under high energy, you don't want to run pyruvate dehydrogenase complex, so it would be in the inactive phosphorylated form. So she could ask about that. Which of the following statements explain why acetate wouldn't be potentially? So I explained this one for 7C. Acetate wouldn't be a product because of you make the thioester high energy bond, and if acetate is released, that means you broke the bond, which is the whole point of the complex, is to make the high energy bond and transfer it into acetyl-CoA. So if acetate is released, that means you broke that bond. And I said I put very important could potentially be open response, because if I was a teacher, I would probably ask this as an open response question. Okay. You guys have any questions?
Question real quick?
Yep, go for it.
There are like multiple enzymes that I would say have like some sort of phosphorylation or dephosphorylation to be activated or inactivated. Would you suggest like memorizing all of those and like all of the enzymes that do that activating and inactivating, or do you think that's like too in depth for what she wants?
I think you should know it.
You said this one, but like obviously there's more than just this one, and I feel like they're all low-key kind of random, so I wasn't sure if that's like too much or not.
Okay, so an easy way I would remember it, usually a phosphorylation activates an enzyme, but pyruvate dehydrogenase complex is one of the few that are inactivated when it has a phosphate. So that's one way to remember if a phosphate activates or deactivates. Usually they're just called the enzyme name with a kinase and then the enzyme name with a phosphatase. I would know them, but if you don't have the time, focus on other important stuff, like big picture and stuff.
Thank you.
Yep. So like, for example, if you have no idea what phosphorylates pyruvate dehydrogenase complex, it's most of the time it's going to be pyruvate dehydrogenase complex kinase. And if you have no idea what removes the phosphate, it will be pyruvate dehydrogenase complex phosphatase. So most of the time, the name tells you what the enzyme does. Okay, what is this? This is talking about substrate cycling between PFK and fructose 1,6-bisphosphate. And then they want to know if this cycle is a control point for glycolysis in the liver or not. So describe how the cycle works, how it's regulated, and how it controls flux through each of the pathways. I think it's easier if I just pull a picture of this. I'll be back here. Is it this one? Yep, this is. I think this is the best picture to go off. So PFK, the main regulatory enzyme in glycolysis. So this is glycolysis. Glycolysis would want to run fructose 6-phosphate to fructose 1,6-bisphosphate, and then fructose 1,6-bisphosphatase runs the opposite. It also uses water and releases a phosphate. This one uses ATP and releases ADP. So between them, remember PFK is regulated by a lot of stuff. It can be regulated by ATP, AMP, oxygen levels, proton concentration. It can be regulated by PFK2, regulated by a lot of stuff. But usually if you have high ATP, you have high energy. You have a lot of energy, so you don't want to run glycolysis. You don't want to make ATP. So you will inhibit glycolysis, and you will activate fructose 1,6-bisphosphate. If you have high AMP, so AMP is low energy. So if we have low energy, we would want to run glycolysis. So this would activate glycolysis. And we don't want to run gluconeogenesis when you have low energy, so we would inhibit fructose 1,6-bisphosphatase. And then fructose 2,6-bisphosphate, so this is made by PFK2. So PFK2's whole job is to make this. And fructose 2,6-bisphosphate activates PFK and it inhibits fructose 1,6-bisphosphatase. Okay, so that's the main way they're regulated together. 8B in the fructose 1,6-bisphosphatase were insensitive to AMP, predict what would happen. So she could ask anything about that, but you would have to understand how AMP regulates fructose 1,6-bisphosphatase to answer this question. So remember, AMP is low energy, so it would inhibit fructose 1,6-bisphosphatase. But if it's insensitive to that, it will not be able to inhibit it, and you would have to talk about what causes would the cell have to go through. So for example, if it's insensitive, I think she said the gluconeogenesis would increase even when we don't want to run gluconeogenesis. I have questions you could ask. So she could tell you under high energy conditions, how are PFK and fructose 1,6-bisphosphatase regulated? So high energy conditions, you would have ATP, and you would have to say that ATP inhibits PFK and then activates fructose 1,6-bisphosphatase. She could also switch around. She would be like under low energy conditions. So that's why it's important to understand the concept and not just try to memorize these questions straight from the solutions she gave you. For AB, which of the following explains what would happen if PFK is insensitive to ATP? So this is similar to that, but instead of asking what would happen if fructose 1,6-bisphosphatase was insensitive to AMP, you want to be able to answer it with any kind of enzyme. So the one I gave here is if PFK is insensitive to ATP. So if PFK is insensitive to ATP, you would still run glycolysis under high energy levels, and you will not be able to run gluconeogenesis as efficiently when you have like low blood glucose levels, for example. And then I have the regulations for both of them because I think there might be a question on that. Okay, so she gives you, this is talking about NAD and NADH ratios and how this can regulate many metabolic pathways in the cell. Indicate the major function of each of the enzymes and indicate how changing the NAD and NADH ratio can change the outcome. So this is another reason why it's important to know all the TCA enzymes and the glycolysis enzymes. This is usually from glycolysis, lactate dehydrogenase. Glycerol 3-phosphate dehydrogenase, that's from the glycerol 3-phosphate shuttle. And it's also in glycolysis, like you learn it with glycolysis. This is in TCA, alpha-ketoglutarate dehydrogenase. And then PepCK, that would be gluconeogenesis. So the first part of this is you would have to say what the enzyme does and what it catalyzes. And when you know what the enzyme does and catalyzes, you'll be able to answer the questions based on if the NAD-NADH ratio is changed. So I'll just do one example of these. So for lactate dehydrogenase, just to show you how it would work. So if we have pyruvate plus NADH, this goes into lactate plus NAD. So basically, let's say, for example, they tell you that you have a high NAD and NADH ratio. How would that affect lactate dehydrogenase? So if we have high NAD, this would shift this way. So we would have more pyruvate than lactate. And then you can talk about what this pyruvate would do. So you can be like, pyruvate goes into TCA, goes into pyruvate dehydrogenase complex, which eventually goes into TCA and make electron carriers and increase the energy levels of the cell. So that's how you would answer these questions. So knowing where NAD and NADH are in the reaction will help you answer this. Okay, so I think that happens for all of them, but I'm not going to go over all of them because this is taking longer than I expected. Okay, that's a really long question. For each of the following conditions, describe the normal function of the enzyme in detail and how the indicated modification will induce pathology. Okay, so the first one, she wants biotin. and pyruvate carboxylase. So I think I have it here. Whenever you see biotin, think of addition of CO2. Biotin is the go-to cofactor when you're trying to carboxylate something. So biotin, I have the mechanism over it, but I can't remember it right now. But it basically helps transfer the CO2 from bicarbonate to pyruvate. So that's the main function of biotin and pyruvate carboxylase. And I think she has pyruvate carboxylase catalyzes the first reaction in gluconeogenesis. So pyruvate carboxylase, remember, goes from pyruvate to oxaloacetate. And it uses biotin to help with the carboxylation. She says the CO2 comes from bicarbonate. And then you make the flexible arm. I don't think you have to know it in that much detail. The enzyme undergoes a conformational change, and the CO2 is eventually bound to pyruvate, and then you would eventually make oxaloacetate. Okay. So then for 8b, she says hexokinase in the brain has a mutated to have a Km equal to glucokinase. Okay. So I think I just have a question on this. So remember glucokinase is in the liver, and it has a very high Km. And then let's just talk about the brain for now. So brain has a hexokinase with a low Km. Km just tells the cell how likely they are to bind to a certain substrate. So low Km requires very low concentrations for the reaction to run. So the brain, because it runs mainly on glucose, has a low Km because it binds with high affinity all the glucose in the blood. The liver wants to have a high Km because the liver wants to give other organs that have higher priority the glucose in the blood. So that's why it has a higher Km, low affinity. And the brain has low Km, high affinity for glucose. So they're telling you what happens if the brain is mutated to have a hexokinase with a Km similar to that in the liver. Basically, what will happen is the brain won't get sufficient glucose from the blood. And you can talk about what pathologies that could cause. So I think she explains what hexokinase does. So hexokinase takes glucose and converts into glucose 6-phosphate. It's the first regulatory step in glycolysis. And then she mentioned the Km briefly. She says the Km for glucokinase is 100 times, meaning that it has a low affinity for glucose, right? So that's just what I explained. And then it says glucokinase is not expressed in the brain cells. So you don't have to worry about competition. If hexokinase had a larger Km, flux through glycolysis would be slower and the brain would receive less glucose. Okay, so that's all of that question. Make sure you're able to answer that in a different context as well. And it doesn't just apply to glucokinase and hexokinase. This is just one example in your body. A lack of malate dehydrogenase in the cytosol of hepatocytes. So this is malate dehydrogenase in the so malate dehydrogenase in the matrix is for TCA. Malate dehydrogenase in the cytosol is for the malate aspartate shuttle. So if we have a lack of malate dehydrogenase in the cytosol, our malate aspartate shuttle won't run, so we won't be able to make enough NAD for glycolysis to run. I think that's what she has as well. Malate dehydrogenase catalyzes the formation of oxaloacetate from malate and vice versa. During gluconeogenesis in hepatocyte, oxaloacetate generated by pyruvate cannot cross the membrane. It must first be converted to malate. So this is what she's talking about with the malate aspartate shuttle. I think it's easier if I just pull it up. I like pictures more than like the long paragraph she has. Okay. So she's talking about, so this is the cytosol, this is the matrix. So malate dehydrogenase. So this is the one in the cytosol. So this is your TCA1. This is the one in cytosol. So if the ones in the cytosol is not running, we won't be able to make malate here, and we also won't be able to make NAD. This NAD in the cytosol is used by GAPDH for glycolysis to run. If we're not making any D-inositazole, this will slow down glycolysis. And if you just say that for that question, that will show enough understanding. And this is the malate aspartate shuttle, by the way. Okay, I don't think she talks about the malate aspartate shuttle. In the cytosol, says I can access malate. If there were no cytosol, I can say solid state would decrease. Yeah, I'm not sure. I had this question on my test last year, and Dr. Shriver wanted you to talk about the malate aspartate shuttle, and I think that's what this question is hinting at. For 9D, pyruvate kinase is insensitive to fructose 1,6 bisphosphate in the muscle. Okay, so this is the concept of feed-forward activation. So remember, whenever PFK is running really fast, you make a lot of fructose 1,6 bisphosphate. That can, so let's just draw it here. So PFK, if PFK is running really fast, you make a bunch of fructose 1,6 bisphosphate. This can go down. It's something called feed-forward activation and activate pyruvate kinase, which is the last step in glycolysis. Okay, so that's what this one is talking about. Yeah, I think that's all she says. If it's insensitive, you won't have the feed-forward activation and glycolysis would run slower. And then for E, she has red blood cells that are deficient in 2,3-BPG. Remember, 2,3-BPG mutase catalyzes 2,3-BPG mutase makes 2,3-PPG, and 2,3-BPG increases the affinity for oxygen. So if we have a deficiency in 2,3-BPG, we won't be able to make enough 2,3-BPG, and red blood cells will carry less oxygen. And I think that's what the explanation says. Okay, so what she could ask about this? So she could ask which cofactor is commonly used in carboxylation reactions. So you would say biotin for that. Which of the following scenarios would occur from a hexokinase having Km? So she could give you different scenarios, and you would have to pick which one would happen. If hexokinase in the brain has high Km, so you would pretty much pick the one that says glycolysis is running slower. Which of the following conditions is explained by a deficiency in isocitrate dehydrogenase? So I think I said a lack of malate, because she asked a lack of malate dehydrogenase in the cytosol. Be able to answer a question based on any deficiency of any of the enzymes in TCA. 9D, which substrate is involved in a mechanism known as feed-forward activation? So 9D, remember, this is fructose 1,6 bisphosphate is involved in a mechanism by activating pyruvate kinase called feed-forward activation. So she might ask you which substrate is involved in a mechanism known as feed-forward activation. So that would be fructose 1,6 bisphosphate. And then she could, for 9E, she could ask a deficiency in 2,3-BPG leads to which of the following in red blood cells? So a deficiency in 2,3-BPG would lead to less oxygen being carried. If you guys have a better questions that you can come up with from the problem sets, feel free to. I kind of just made these really quickly when I was going through it. Okay. What is this? So rapid flux through glycolysis is a hallmark of cancer cells. And they're giving you, okay, so you have cancer cells and they're trying to make treatments. So they want to know if these enzymes would be good targets for inhibition. Okay? So you have to first say what the substrates and the products of the reaction are. You would have to indicate whether the enzyme is reversible or irreversible. And based on your answers from A and B, justify if the enzyme is a good target or not. Okay? So lactate dehydrogenase. So remember, lactate dehydrogenase takes pyruvate, converts it into lactate. So this is if you're running anaerobically. So this would happen a lot in the muscle. So for part B, indicate if it's regulated and what are the regulatory mechanism for it. It's not regulated. It's only regulated by levels of pyruvate, lactate, or NAD and NADH. But other than that, it's not regulated by anything else outside the cell. Also, if they're asking you a question on that, if the enzyme is not regulated, it's probably not a good target for inhibition. So this is probably not a good target, lactate dehydrogenase, because it's at the end of glycolysis. It's not a regulated enzyme, and it's very reversible. The next one is PFK. So this is the committed step. Committed steps are really good for to be targets for inhibition when you're trying to stop a pathway. So PFK, remember, converts fructose 6-phosphate to fructose 1,6-bisphosphate. And it's the major regulatory point. It's regulated by like 6 different things. So this would be a really good target for inhibition, PFK. The last one is GAPDH. So this one is tricky. So GAPDH, remember, converts the oxidation of GAP. And then remember, it's a step that uses NAD. And it makes 1,3-BPG. Okay, so this enzyme is reversible, but one, it uses NAD. So, and NAD is a really big factor of in glycolysis. So if you inhibit the only enzyme that uses an NAD, glycolysis will not run. And 2, although the enzyme is reversible, our 2 substrates, so we take GAP and we make 1,3-BPG. The enzyme is reversible. It has a 0 negative delta G, but this product is high energy, and this one is low energy. And the reason it's reversible is if you look at, I explained the mechanism of it in the video I have. If you look at the mechanism of it, you have this intermediate that you make inside the enzyme that is very high energy. So it allows the enzyme to be reversible while still making a product that's a high energy product. So it's even though the enzyme is reversible, it would still be a good target for inhibition. So some questions you could ask. Which of the following enzymes would be a good target enzyme to inhibit in cancer cells when they're running glycolysis? So you would pick if she gives you a list of enzymes, you would pick the ones that are the main regulatory enzymes. For 10b, she could ask, explain in full detail what PFK catalyzes. So if you know the reactions in glycolysis, this shouldn't be a problem. And you would have to indicate if it's a good target to inhibit. So PFK is always a good target to inhibit. And for 10c, I have a question. GAPDH is a reversible enzyme. Which of the following explains why it's still a good target for inhibition? So because it's the only enzyme that uses NAD, and because it makes a high energy product from a low energy substrate, it's still considered a good target and enzyme to inhibit. Okay. I might have to go a little bit faster if we want to go over everything because we're only on question 11. Passive effect is important for regulation of glycolysis when oxidative metabolism is occurring. So within a rapidly growing tumor, there is often inadequate oxygen. Based on your knowledge of the Pasteur effect, so explain the labeling. I thought about making a labeling video, and I think I might this weekend because I think labeling it can be confusing. So you would have to say, if you give the cell 13C glucose, you would have to explain what would be labeled, what metabolites would be labeled. And the condition you're given, it's a rapidly growing tumor cell. So let me explain labeling real quick. So if you have a cell, all the glucose inside the cell is 12C. So that's glucose that it made from previous cycles. What labeling does is you give the cells 13C something, and it allows you to track which pathways are being upregulated. So whichever pathways have the 13C are the pathways that are being upregulated. And whichever pathways still have 12C are the pathways that are still running at the same level, or they're downregulated or whatever. So labeling just allows you to track which pathways are being upregulated. So that's the whole point of labeling. So they want to, with inadequate oxygen, you want to know how these substrates will be labeled in TCA and glycolysis. So with inadequate oxygen, pyruvate will be reduced by lactate. So remember, in inadequate oxygen, you run anaerobically, so pyruvate is converted to lactate. So pyruvate and lactate are in equilibrium with each other. Pyruvate doesn't go into pyruvate dehydrogenase complex. So all the 13C carbons that are given in the cell are going to be in glycolysis, in lactate, but they will not be in TCA. They will not be in TCA because you won't be running TCA or ETC when you don't have enough oxygen levels. So your glycolysis would be upregulated, so they would be labeled. Lactate would be upregulated, so it would be labeled. And then TCA would not occur, so you would not see any labeling in TCA. And if you have a perfused tumor with oxygenated blood, would the labeling pattern change? So now it's the same tumor, but now you're given it oxygen, and they're asking you if the labeling would change. So of course it would change, because now the cell can run TCA. So glycolysis will still be labeled, so glycolysis won't be affected by oxygen levels. But now, because we have oxygen levels, we can run aerobic respiration instead of anaerobically. So lactate will not be labeled, and then TCA intermediates would be labeled for that one. Okay. Some questions you could ask: under high oxygen levels, which of the following enzymes would be upregulated? And they could also be applied to labeling. So you could be like, you give them 13C, which ones would be labeled? Which ones would be labeled is another way for which enzymes are being upregulated at the current moment. So under high oxygen levels, glycolysis, TCA would be upregulated. You will not run anaerobically. Let's see. Those are the only two for sure that would run. If you're under high energy, you would also run glycogen synthesis. 11B, a question she could ask: if 13C glucose is fed into tumor cells, out of the following substrates, which would you expect to be labeled? So you have to know that tumor cells do a lot of glycolysis. They divide all the time, and then they mainly run on glycolysis. So this is actually a treatment. A lot of times you want to know where tumor cells is, you check where 13C glucose is, because they do a lot of glycolysis. So they could give you a bunch of different substrates, and you would have to pick the ones in glycolysis for that kind of question. Okay. I don't think I included questions on this one. Okay, draw the TCA reaction that is catalyzed by the enzyme. Oh yeah, because for this you just have to know. Now I draw the TCA reaction that is catalyzed by the enzyme succinyl-CoA synthase. So succinyl-CoA synthase takes succinyl-CoA and makes succinate. So this is in TCA. The delta G of this reaction is approximately 0. Why is this reaction reversible despite substrate level phosphorylation? So this is really similar to how GAPDH works as well. Is you use substrate level phosphorylation, but the enzyme level is well, you don't use substrate level phosphorylation in GAPDH, but it's a similar concept. Basically, use substrate level phosphorylation, but your products that you make are let's see what she said. Both forward and reverse reactions involve the cleavage of a high energy bond. The forward reaction involves cleaving of a sulfide carbon bond, whereas the reverse cleaving of a phosphate bond. I don't think she really maintains the high energy bond, considering the chemical energy. I don't think she really explains it, but basically because you're two, I think, let's just go back here. I have it here. Yeah, so we make the phosphate attack succinyl-CoA, you make succinyl phosphate, and then eventually you make this high energy intermediate is why this enzyme is reversible. We're able to transfer this energy into GTP at the end instead of making a high energy substrate. So we have phosphate, succinyl-CoA. We make succinyl phosphate. And then we make this high energy intermediate. So instead of giving succinate the high energy and making the reaction irreversible, this bond is broken, and eventually this high energy phosphate is given to GTP. And then you make another different high energy product, which is GTP. So you make GTP and then maintain succinate at a very similar energy level as succinyl-CoA. So that's why this one would not be irreversible. Okay, what would happen to the overall delta G of this reaction in TCA flux in general if succinyl-CoA is pulled out of TCA? So I think for this you have to know not just for succinyl-CoA, but basically what happens if any of the substrates are pulled out. So succinyl-CoA is right here, and it goes into succinate. So if you pull succinyl-CoA out, this will shift. This will make succinyl-CoA the low concentration. So which the two reactions on succinyl-CoA would both favor succinyl-CoA. So succinyl-CoA synthase would favor the succinyl-CoA direction. And the enzyme right before succinyl-CoA, which is alpha-ketoglutarate dehydrogenase, that will favor succinyl-CoA even more. So that's similar to what would happen if you pull out a substrate out of any of the cycle, out of any pathway, sorry. What would happen to the flux if the histidine active site is mutated to alanine? So actually go back. This is a histidine residue. So you need histidine for this reaction to occur. If histidine is not there, the reaction won't happen. So if you do alanine instead, the reaction will not happen. Okay. 13. This is taking a lot longer than I thought. So this metabolite is produced by macrophages by the enzyme cis-aconitase decarboxylase. And this is the structure for it. And they're telling you it's produced by a simple decarboxylase reaction. You have to predict the substrate used by the decarboxylation reactions that produce it. So you don't really know this enzyme, and it's kind of hypothetical. So they give you this structure called, and they don't tell you, it's called itaconate. I don't know how to pronounce it. And they tell you it's catalyzed by, actually they don't, they tell you, sorry, it's catalyzed by cis-aconitase decarboxylase. So because it's catalyzed based on the enzyme name, this is not one of the enzymes you guys have to know, but based on the name of the enzyme, they expect you to know that it takes cis-aconitase, which is an intermediate between aconitase and isocitrate. And you would have to explain what would happen if the reaction is upregulated in the product, the substrates for it, and you have to explain your logic. So I think she said since the enzyme is called cis-aconitase decarboxylase, the substrate is likely cis-aconitase, and then because it's a decarboxylase, she says it would pretty much involve a decarboxylation reaction. I don't think you'll get asked on this because I don't remember any stuff on this stuff. It says in some immune cells they produce aconitate, all glucose is consumed and excreted out of the cell as lactate. Explain why this would occur. So I think, let's see, in some immune cells they produce all glucose consumed, which is an immune isomerization reaction. Reaction to produce. I think all of this is asking is if you have a substrate that is So for example, the example I gave here is with alpha-ketoglutarate dehydrogenase. So alpha-ketoglutarate can be converted into glutamate, and it can also be converted into succinyl-CoA. So the whole concept she's testing is if she's telling you that if alpha-ketoglutarate is going all into glutamate, what happens to succinyl-CoA levels? And you would pretty much have to say that they drop. So that's all the concept she's testing with this. She uses enzymes and substrates that you guys are not expected to know. I don't think she will test you over this at all. Okay. Question 14. So they give us this compound DFX, and they tell us it disrupts oxidative phosphorylation. So this is just ETC. And they tell you it does this by sequestering iron, and then you treat hepatocytes with this compound or a negative control. So our negative control in this case is hypoxia. The reason hypoxia is a negative control is because if something disrupts oxidative phosphorylation, you would expect it to act the same way as hypoxia. Okay, so this is kind of what you would expect this compound DFX to act like. So that's why I use hypoxia as a negative control. So you treat hepatocytes with this compound or a negative control and measure the amount of induction of a gene controlled by HIF1. So you pretty much give two different cell lines DFX, two different cell lines. One of them is treated with DFX and the other one is hypoxia. And you measure, using a Western blot, you measure this protein and you also measure actin. Actin is usually used as a control. It allows to load and control to make sure that you did your experiment right. So explain how DFX would change the activity of the protein HIF1 below. So basically after we added DFX, we see that there is an increase in HIF1. So you can, if you're given a similar graph on the test, you can talk about it can be any kind of protein, but the first thing you can say is the thing we added increased HIF1. When you guys did this homework set, you guys didn't know what HIF1 does, but now you do. It's called hypoxia inducible factor, so it upregulates a lot of stuff that happened during hypoxia. So one of them would be glycolysis. So is a master regulator among other things that induces transcription of genes in glycolysis, and it increases gene transcription of HIF1 through. And then she talked about this through stabilization. I think this is from the book on how it increases transcription of glycolysis. And then for each of the following, explains how DFX would change the regulation. So basically, what this question is trying to ask is they gave the cells these things that inhibit oxidative phosphorylation, you inhibit ETC, so you're not making ATP. So she wants to know what happens when you have low ATP levels. So that's the whole point of this question, really. So the first thing that will happen, glucokinase would be upregulated because you're in a low energy state and you need to make enough ATP. Malate aspartate shuttle makes NAD in the cytosol, so that would be upregulated as well. Pyruvate kinase, that's an enzyme in the last enzyme in glycolysis, so that would be upregulated. Succinate dehydrogenase, so that's in TCA, and that usually runs when ETC is running, so that would be downregulated. And then PFK is the main enzyme in glycolysis, so that would be upregulated as well. So for this, you want to be able to interpret Western blot and any kind of graphs, and then select. You could select all of the following enzymes that would be upregulated under hypoxic conditions. So she will give you a list of enzymes, and you would be able to pick which one would be upregulated under hypoxic conditions. So that's something I could see her asking from this question. Okay, we'll finish problem set 1, and then for the other things, I think, unless no one has questions, we'll go over them. but it will take too long going over every single question in the problem sets, I feel like. So we have this glycogen storage disease, and it's caused by a mutation in phosphorylase enzymes in the muscle. The symptoms are muscle cramps and pain, fatigue, and basically they do this test. This test measures lactate in the blood, and they want to know why lactate in the blood is a good way to diagnose these patients. Okay, so basically, if these patients can't break down their glycogen, they won't be able to make glucose. So flux through glucose would be low, and then you won't make enough lactate. So lactate levels would be low. So that's why you could use lactate levels as a way to diagnose this patient. For part B, 15B, patient is undergoing ischemic exercise, and then so it's the same thing. They're fed 13C glucose, and the blood is drawn after the test. Explain how the following substrates would be labeled. So ischemic exercise, if you ever see that, it's really similar to like hypoxic conditions, I guess. Ischemia is lack of blood flow, and if you don't have blood flow, you'll have lack of oxygen. So alpha-ketoglutarate, that's in TCA intermediate. So under ischemic conditions, you will not run TCA, so that it will not be labeled. Glucose, again, glycolysis and glucose, those don't matter. They don't depend much on oxygen levels, so those will still run. Ribose, that one is in the pentosphosphate pathway, so that one can be labeled. Not really. You can't really say if ribose will be labeled or not, so I think that's why she has maybe activated, and it can be labeled. Alanine. So alanine is made from pyruvate, one of the reactions that alanine is made of. So that one will not be labeled because most of your pyruvate will be going through lactate. And then for C, how would you expect the electron transport chain to increase, decrease, or remain the same? So at rest, the electron transport chain would remain the same because remember, these are muscle cells, and muscle cells mainly rely on glycolysis mainly to make their energy. So at rest, this won't change. And then during exercising, she has decrease. She has higher ATP consumption, needs more electron transport chain and oxygen. However, need to release glucose stores through glycogen breakdown. So basically, I think it's really confusing how she worded this. Basically, she's saying if you break down glycogen into glucose, you would expect that the higher ATP consumption during exercising would need more electron transport chain, but because you can't break down glucose in this patient because they have a phosphorylase deficiency, ETC activity would decrease. I think if you also put stays the same, that won't be wrong as well. I think then I have some questions you could ask. So for 15A, so in a deficiency of phosphorylase enzyme in the liver, which of the following explains whether lactate dehydrogenase is downregulated? So that would be the explanation for 15A. So she would give you like different scenarios, and you have to pick which one explains why lactate dehydrogenase is downregulated. So you would have to say that glycogen is not being broken down into glucose, and that's not going into glycolysis, and you're not making lactate. 15B, she could ask under ischemic conditions. So ischemic conditions very similar to hypoxia. Which substrate would you expect to not be labeled? So she would probably give you glycolysis intermediates, and then she would give you one from like TCA or something, and the answer would be the option from TCA, because under ischemic conditions, intermediates from TCA would not be labeled. 15C, which of the following explains why ETC is downregulated if glycogen phosphorylase is inhibited when exercising? So that would be because we're unable to break down glycogen into glucose, so we're not making enough electron carriers for ETC, so then ETC is downregulated. Okay, I think for this is talking about dihydrolipoamide deficiencies. Remember, lipoamides in pyruvate dehydrogenase complex and alpha-ketoglutarate dehydrogenase. And you have to, so DLD catalyzes the production of lipoamide. So lipoamide is used in E2 of alpha-ketoglutarate dehydrogenase and pyruvate dehydrogenase complex. So you have to describe the catalytic mechanism in detail. So that would be, you'd have to talk about E1, E2, and E3 for that, which we went over at the beginning. Muscle cells from these patients are cultured with uniformly labeled 13C glucose. Predict the labeling pattern. So that's another labeling question. So if this patient has, here, let me see if I can get an open page. Let's go here. So if this patient has a lipoamide deficiency, we will not be able to go from pyruvate to acetyl-CoA. So the stuff down here would not be labeled. Anything above here would be labeled. So that's how you would answer these labeling questions with a deficiency. You'd have to say where the deficiency is. So for example, fructose 6-phosphate, that's in glycolysis, that's labeled. PEP, that's in glycolysis, that's labeled. Pyruvate in glycolysis labeled. So remember, citrate now, that's below. So citrate is down here. It's actually right here. Citrate is here, so citrate would not be labeled. Alpha-ketoglutarate, that's in TCA as well, so that's not labeled. Succinate in TCA as well, that's not labeled. Malate is not in, I think she says possibly labeled because she talked about the reaction of pyruvate carboxylase going into oxaloacetate. So that's why she said possibly labeled for all of these. And then aspartate, she said possibly labeled for this one as well. Okay, predict the following pattern in the muscle cells in the same intermediates if given labeled 13C. So this is different. So now you're given the cells 13C instead of 13C malate instead of 13C glucose. So your entry point is just different. So remember, we have a deficiency here in the lipoamide arm. So now that we're entering here with malate would be labeled, oxaloacetate would be labeled. All of our acetyl-CoA is coming from other parts of the cell. It's not coming from pyruvate dehydrogenase complex. So acetyl-CoA would not be labeled, but citrate would be labeled using different color pairs here. Citrate would be labeled, and then you would go through the cycle again. You also have to remember that alpha-ketoglutarate dehydrogenase is not running because alpha-ketoglutarate dehydrogenase uses lipoamide as well. Okay, so that's how you would answer these labeling questions. You would just have to see where the deficiency is and track which substrates are being labeled and which ones are not. Okay, yeah, I think I have for 13C and a deficiency of DLD, which of the following would you expect to be labeled? So she could give you the same deficiency. So if it's a deficiency in DLD, you would have to say which ones are labeled and which ones are not. And then you want to be able to do this the same with 13C malate or any other substrate. Okay. And I think the last one I have is 17. Okay. Pyruvate dehydrogenase phosphatase. So this is a pyruvate dehydrogenase phosphatase deficiency, and you pretty much just have to talk about it. So pyruvate dehydrogenase phosphatase, remember, it activates pyruvate dehydrogenase complex and puts it in the active form. So if you have a deficiency in that, pyruvate dehydrogenase complex is going to be in the inactive form the whole time. So it will run a lot less efficiently if you have a deficiency in that enzyme. So that's all she talks about here. And then she says, imagine a patient consuming a lot of pasta. So that would be a lot of carbohydrates, so glucose. and then you would have to say if glycolysis would run faster, slower, or stay the same. She said glycolysis would increase because you would have, if pyruvate dehydrogenase complex is not running, you will have less ATP. So this would activate PFK, and then glycolysis would run faster. And then for part B, she says explain normal functions of the underlying molecules of proteins below. Indicate whether you think the following situations might alleviate the pathology seen in deficient patients. So supplementing patients with lipoamide. So lipoamide is a cofactor that probably won't help the patient run. So remember, lipoamide is a cofactor in E2. So that one will not help, and I think that's what she explained. And then she said, given the patients an inhibitor of pyruvate dehydrogenase kinase. Okay, so that would be inhibiting an inhibitor. Pyruvate dehydrogenase kinase is an inhibitor of pyruvate dehydrogenase complex. So that one would actually help. Given the patient biotin. Biotin is a cofactor for pyruvate carboxylase. So because, here, let me draw it. Pyruvate can go into acetyl-CoA or oxaloacetate, and that's how they enter into TCA. So if this is inhibited, she's saying if we give it biotin, so we're activating this pathway. So she said if there's enough acetyl-CoA, it would actually help. So if there's another source of the cell that's making acetyl-CoA, given biotin would actually help. Okay, increasing the activity of the enzyme isocitrate dehydrogenase. I don't think that would help at all because isocitrate dehydrogenase is like down here. So that wouldn't help at all. Okay, so that's what I have for problem set 1. A question she could ask is select all of the following that could potentially be a treatment for a patient with pyruvate dehydrogenase complex phosphorylase deficiency. So you would have to, so remember the only one we said that would be a good example would be the pyruvate dehydrogenase kinase inhibitor. Other than that, we said the other ones will not help. So if she has the same question, that would be the answer. If not, if it's a different scenario, you would have to go through it the same way we did with that. Okay. That's what I have for problem set 1. I think it will take too long to go over every single question because I have problem set 2 as well and problem set 3. So I think what we'll do instead is if you guys have questions on problem set 2 and problem set 3, we'll specifically go over them. Or if you have questions in general about any of the pathways, we can go over that. But it would take like 4 hours to go over everything. So I will be on until like 9:30 taking questions.
Manuel.
Yes.
Can you release what you have for the problem sets?
Yes. I think what I have is I don't have explanations really. I have questions that she could ask from them. Does that make sense?
Yeah.
Okay, but I'll still try to find a way to release it and put it on Brightspace. Is there any questions on problem set 2?
Manuel, I have a question.
Yep.
This might be a dumb question, so
no, there's no dumb question.
But I was looking at gluconeogenesis, and I was talking to you about this earlier, but it's like to start off, you always start with pyruvate, and that pyruvate like goes to oxaloacetate.
Yep.
So when I was like looking at how it all connects, like pyruvate usually goes into the TCA cycle and like through all those steps to make oxaloacetate. If you can just use pyruvate carboxylase to go from pyruvate to oxaloacetate, why does it go through all those steps of the TCA cycle? I mean, I get that it makes like the electron carriers for electron transport chain, but like, why does it sometimes just use that one enzyme and sometimes go through like all those steps?
You're talking why TCA uses pyruvate carboxylase? Is that what you're asking?
No, I think she's asking like if we can go directly from pyruvate to oxaloacetate, then why don't we do instead of using like the 50,000 enzymes in the TCA cycle to achieve the same thing?
Yeah.
Oh, well, you want to get your electron carriers. How do you think ETC makes all the energy from?
If you yeah, that's what I said, but I wasn't sure if there was another reason.
Yeah, the whole point of pyruvate carboxylase in TCA is if you have really high levels of acetyl-CoA and you have low levels of oxaloacetate, you'll downregulate pyruvate dehydrogenase, and you'll upregulate pyruvate carboxylase to compensate for that. So that both of them, because oxaloacetate is the rate-limiting reagent in citrate synthase right here. So if we have low levels of oxaloacetate, it doesn't matter how much acetyl-CoA we have. So the whole point of pyruvate carboxylase is to make oxaloacetate for citrate synthase to run, but the whole TCA is not just to make oxaloacetate at the end. It's a cycle. TCA oxaloacetate is not the end of the cycle. You go through the cycle to make all these electron carriers to be able to go into ETC and make energy. Does that make sense?
Yeah, so then how does your body know like when to take pyruvate and like do like pyruvate dehydrogenase to go through the TCA cycle versus like using the pyruvate carboxylase just to go straight to oxaloacetate?
It will depend on the levels of acetyl-CoA and oxaloacetate in the cell. So if you have high acetyl-CoA and low oxaloacetate, we'll favor this reaction if we want to run TCA. And if it's the other way around, so if we have high oxaloacetate, you're never going to have high oxaloacetate in the cell because oxaloacetate is usually at low concentrations. And low acetyl-CoA, the cell will favor this direction more and convert pyruvate into acetyl-CoA. But that's the whole point of pyruvate carboxylase is just to make enough oxaloacetate to continue TCA. whenever oxaloacetate is in low concentrations. Does that answer it? Your question?
Yeah, that makes sense. Thanks.
Mm-hmm. So is pyruvate carboxylase favored then when we're running gluconeogenesis?
Yes, pyruvate carboxylase is favored when you're running gluconeogenesis. It's the same enzyme, so basically we want to go from pyruvate to PEP, but there's no enzyme that does that, okay? So we can't do that. So we do a different pathway. We convert pyruvate to oxaloacetate, and then we convert oxaloacetate back into PEP. So instead of, because there's no enzyme that can convert pyruvate to PEP right away, we convert to oxaloacetate, and then PEP-CK allows us to convert between oxaloacetate into PEP. So that one overpasses the enzyme right here, which is pyruvate kinase. So we overpass pyruvate kinase by doing this loop.
And is there any regulation specifically on pyruvate carboxylase that like causes it to be upregulated when in hypoglycemic conditions, or like what is the regulation that causes that to be favored in oxaloacetate? And then in gluconeogenesis, sorry.
Under hypoglycemic conditions, it will not be upregulated. It will be downregulated. So you mean like under hyper?
No, hypo, like low blood glucose.
You don't want to run pyruvate kinase under low blood glucose levels.
No, pyruvate carboxylase.
Oh, pyruvate carboxylase. Sorry. There isn't. It will mainly have to do with insulin and glucagon. So the first thing they will do is inhibit this one. There's a lot of regulation on this one. You will inhibit PFK, of course. And any pyruvate that you have in your cell will be converted into glucose, so it's released in the blood. So you just inhibit this one. Remember, pyruvate dehydrogenase complex has a lot of regulations, but I don't think pyruvate carboxylase is regulated by much. I know it needs acetyl-CoA to run. But other than that, I can't think of anything. Wait, go ahead.
I just said that makes sense. Thank you.
Okay. Sounds good. Is there any other questions, or would you guys prefer if I just ask you guys questions on different pathways to test like your knowledge?
I have a couple more questions.
Okay.
Okay. That would be awesome.
We can.
Okay, I saw it. You passed this slide actually earlier. I was just wondering, where is NADP dependent isocitrate dehydrogenase located?
It would be in the mitochondrial matrix. This one?
No, the other one, the second line down.
Wait, isocitrate NADP dependent. What? Right?
Yeah, NADP dependent one. I was just wondering. I know where the first one is. I just don't know where the second one is. Cytosol, because that would be, I'm assuming, like something with NADPH, which is pentose phosphate pathway.
I'm actually not sure. I just know it has a different isoform, and it's you'll only run it to maintain glutathione. Glutathione is in the cytosol. So I'm not sure where.
Like a solid guess on the test. Would you say cytosol?
I would say cytosol because glutathione is made in the cytosol, but I don't know for sure.
Okay, and then is there a difference between HIF1 and IF1?
Yeah, there is. So HIF1 is a transcription factor. IF1. So HIF1 is called hypoxia-inducible factor, and it's activated under hypoxic conditions. And it upregulates a lot of things that help during hypoxia. One of the things that it increases transcription of is IF1. IF1 specifically binds to ATP synthase and inhibits it. Does that make sense?
Yeah, that's the thing that like doesn't let you run stuff in reverse, right?
Right.
Or,
IF1 is the one that binds.
Yep.
IF1 is the one that binds ATP synthase. HIF1 increases transcription of IF1. Is there any other questions?
My last one, can you just like go through the pathways that are stimulated by insulin and then the pathways that are stimulated by glucagon? Just kind of like make like a list. I just keep somehow getting them mixed up.
Okay, I thought I didn't. Let's see. Yep, I thought I made it here. Yep, when I was talking about adenylate cyclase. So remember, insulin is released under high blood glucose levels. It will activate receptor tyrosine kinase. Some of the things that it activates are glycogen synthesis. So when you have high blood glucose levels, you want to put that glucose somewhere, so you store it as glycogen. Some of the glucose will also run through glycolysis to make energy. You'll also run, because we have a lot of energy, we'll start to store fatty acids, and we'll also start synthesizing cholesterol. Different organs will do different things. So, for example, cholesterol synthesis that happens in the liver, fatty acid synthesis that happens in adipose tissue in the liver, glycogen synthesis that happens in the liver and muscle cells and astrocytes. All the cells do glycolysis. And then glucagon is released under low blood glucose levels. One of the main things that will upregulate is gluconeogenesis in the liver. So the liver will make glucose. Will make glucose through gluconeogenesis and release that in the blood. We will we'll run through beta oxidation to make energy for the cell, and we'll also break down glycogen to make more glucose for the cell.
And then would ketone bodies would also be on that side?
The glucagon?
On the glucagon side?
Let me see. Yes, I would say so. I can't think of a direct way that glucagon acts on ketone body. But yeah, okay. So there is that confusion with this, because whenever you talk about glucagon, you talk about low blood glucose levels. So this is like the starvation phase. After that, you kind of just have like a fasting phase where it's like an extreme starvation. That's when you start making ketone bodies. So it's a little bit after that when you start making a lot of ketone bodies, and your body starts consuming ketone bodies as their main energy source. Does that make sense?
Yeah, but it's definitely more on like the glucagon side.
Oh yeah.
Okay, just want to make sure.
Yeah, you're not gonna make ketone bodies when you have high insulin levels.
Thank you. That's all I had.
Okay. Is there any other questions, or should?
Yeah, I have a question about that same slide. If you don't want to, sorry.
Oh, did I switch it?
My bad.
No, you're good. Oh, what is it?
Kind of a general question, but are like high blood glucose and low blood glucose, is that at all like analogous to like energy levels, or is it completely unrelated?
For the most part, okay. So high blood glucose levels, you'll run fatty acid synthesis, cholesterol synthesis, you'll do glycogen synthesis, and you will do glycolysis. Okay, so let's compare that to high energy levels. Okay, so under high energy levels, you will run glycogen synthesis, fatty acid synthesis, cholesterol synthesis. The only difference is you will not run glycolysis, because PFK will, under high energy levels, will inhibit. ATP will inhibit PFK under high energy levels. So glycolysis would be the only thing that's not run under high energy levels. Instead, the liver, if we're in the liver, for example, the liver will probably make actually it depends on the blood glucose levels if we're gonna do gluconeogenesis or not. But yeah, so glycolysis would be the only one that's like different from that.
And then the same thing would be with low blood glucose levels. Everything like beta oxidation and glycogen breakdown that would happen at low energy level, or
Yep, that would.
Low energy gluconeogenesis is still more like related to blood glucose levels.
Yeah, gluconeogenesis and glycolysis are related to blood glucose levels, but they have a different way that they run depending on energy state. Okay. It's also important to think of what organ we're in. So, for example, when we talk about glycolysis and gluconeogenesis and how they're regulated, only beta cells of the pancreas and the liver do these two pathways. Okay, so if we're in the muscles, if we're in the muscle tissue or the brain, we won't have to worry about that. Okay, so that's only in the liver when you have to consider how glycolysis and gluconeogenesis are regulated.
Okay, thank you.
Mm-hmm.
You said in what pathways do we not have to worry about it? You said the brain and what else?
Yeah, the brain, the muscle, heart, red blood cells. Pretty much all of those don't run glycolysis, don't run gluconeogenesis. They all run glycolysis. So whenever you're looking at regulation between glycolysis and gluconeogenesis, you will only have to worry about that when you're in the liver.
So basically, everything's like really consistent with insulin and glucagon signaling, except glycolysis and gluconeogenesis, and those you have to take also into consideration the energy levels at that point.
Yes.
Thank you. That makes a lot more sense.
Mm-hmm. Is there any other questions, or should I go through?
I actually have one.
Yeah, go for it.
Kind of along the same pathway as that. Is there any way that we can go over the catabolic pathways and then the anabolic pathways? I have a general outline of what I just all of them because I feel like she keeps pounding us with that.
Okay.
And I think that's like a major thing.
Okay,
We should know.
let's actually look at AMPK. So, because AMPK acts directly on that stuff. So anabolic would be building stuff. Like, stuff is not the most, like, you're just building bigger substrates. And then catabolic is you're breaking down stuff. Okay, so anabolic, for example, would be glycogen synthesis. So it's the first one I think of. It would be fatty acid synthesis, protein synthesis, and gluconeogenesis. Okay, I'm trying to organize this the best way possible. There. Okay, so we have glycogen synthesis. We said protein synthesis. And then fatty acid synthesis. Cholesterol synthesis. And then catabolic. Oh wait, gluconeogenesis is also considered anabolic. Gluconeogenesis. Okay.
Is the PPP also in that or no?
I wouldn't consider it anabolic or catabolic.
She said in class that if you were like trying to think about the PPP in terms of like anabolism and catabolism, to like kind of group it more with the anabolic things because it makes stuff that's used to help make other stuff.
Okay, I guess because you make NADPH with fatty acid synthesis, I'll put /PPP here. Okay. Usually it's not regulated by stuff like that. Glycolysis for catabolic. Glycogen breakdown. TCA, and then the electron transport chain. So we have glycogen breakdown, beta oxidation. Those are the main ones I can think of right now. Do you guys think of other ones?
Would ketogenesis or the urea cycle be here?
So in the liver, ketogenesis would be considered anabolic because you're building stuff. And in other tissues, so for example, let's do muscle, ketone body breakdown. Okay. Let's see. I think that's most of them. You guys did amino acid metabolism too. So that, I guess that would be protein synthesis. So usually this is really it goes along with AMPK because AMPK is, let's go in case you're copying that. AMPK is activated by AMP and it's inhibited by ATP. So high AMP would activate AMPK, and then AMPK inhibits a lot of the anabolic pathways, and it activates a lot of the catabolic pathways because we're in a low energy state. So that's why I like to talk about the slide with AMPK, because AMPK acts on anabolic and catabolic stuff. They have glucose uptake for catabolic, which I guess is considered part of the catabolic reactions. Is there any other questions? Okay, I can do would you guys prefer I go over problem set 2, or would you prefer to end it now, or would you prefer me asking you questions, like quizzing you guys, and you guys try to answer, I guess? Hello?
You should ask us questions.
Ask it.
That's my opinion.
Okay, I guess.
I would second that opinion.
Okay, so I guess the best way to do this is go through the problem sets, and then through the concept, I'll ask you questions, and I have them down here. Okay, so this is in problem set 2, question 1. It's talking about glycogen synthesis. So a question you could get asked is, what type of bond does glycogen synthase catalyzes? Anyone? It's a glycosidic bond, but what kind of bond?
At the risk of sounding stupid, peptide.
No, peptide, that would be linking two amino acids together.
Do you mean like high energy bond, or like something along the lines of that?
No, like it's called like alpha. So if you have alpha 1,4, alpha 1,6, beta 1,4, beta 1,6, out of the 4, which one would it be?
I would say alpha 1,4.
Yep. So glycogen synthesis would catalyze alpha 1,4. And then under high blood glucose levels, how is glycogen synthesis regulated? What happens under high blood glucose levels? Go ahead.
Glycogen synthesis would increase because it's kind of a anabolic process.
Right. So, insulin, high blood glucose levels, you would activate insulin. Insulin would bind through the receptor tyrosine kinase. You would eventually activate a bunch of stuff, and eventually activate PKB, and then PKB acts on an enzyme that inactivates glycogen synthase. So it inhibits the inhibitor of glycogen synthase. So then glycogen synthase is upregulated. Let's see if I have. Yeah, so PKB inactivates glycogen synthase kinase and puts it in the inactive state. So that makes glycogen synthase, the enzyme, in the active state. So that's how glycogen synthase is regulated under high blood glucose levels. Okay, so questions?
Question.
Yep.
So on the exam, like, just I guess this is more of your opinion question. Do you think we being able to just answer like, oh, it does this because it's a catabolic or because it's an anabolic process is enough? Or do we need to know that it activates PKB, which does all that other stuff?
I would know the details, especially for that.
Okay, thank you.
If you remember them. If you don't, there's nothing you can do.
Hey Manuel, as do you suggest we basically know pretty well the signaling video that you put out, basically everything in that video? I feel like we've reviewed that a lot. Like a lot of your questions pertain to that information. So do you suggest that we know most of that, or do you think that's a little too much and too in depth?
Well, which one?
The one you're on right now. Whatever
the signaling one?
Yeah.
I would know. Yeah, I would try to know as much as you can about it, because I will guarantee you'll get asked about insulin and glucagon and what they activate and what they. Maybe not in as much detail as I have on it. I get like if you forgot about the PKB part, then you're still fine. I think you could still get a lot of partial credit. And if you forgot about PKB and what it does, I guarantee you that 95% of the class probably forgot about PKB and what it does. I bet like 20% of the class doesn't even know what PKB is.
Okay. Thank you.
I'm that 20%.
No, I think you guys will be fine. Okay, so for
You've been well.
Yeah, what's up, Anna?
Sorry, I asked you a question before you move on to the next question. The previous question that you asked about glycogen synthase, like what bond it forms, and it being alpha 1,4 bonds. So then, if you were to ask which type of bonds does branching enzyme form, then it would be alpha 1,6, right?
That's right. And remember, debranching breaks alpha 1,6, and phosphorylase breaks alpha 1,4.
And then the phosphorylase, that's a rate limiting step, right?
In breaking down of glycogen, yes, I would say so because it's the one regulated. Yep, it is.
Okay,
if I remember correctly.
Could you remind me the glycogen debranching enzyme that would also be creating a alpha 1,4 linkage? Is that wrong? It might be.
No, it is actually. It is. So what it does is, let's see. I have the picture of it. Is this the branching? Yeah. So basically, the branching does two things. It takes these last couple. So basically, phosphorylase comes in, keeps chopping glucose off, but then phosphorylase can't get to the last 4 glucose residues because of steric hindrance. So the branching comes in, takes 3 glucose, and puts them onto the other strand, and then it catalyzes the breaking down of this alpha 1,6. So yes, you're actually right. It takes these glucose and helps make the alpha 1,4. I don't think she will go that much in detail.
Fair enough. Thank you.
But you're right. You're right. Okay. So question 2. So this is about Le Forest disease. So Le Forest disease, I'm not going to explain all of it because that's taking too long. Is you can't store, you can't break down glucose from glycogen. Okay. So this could be one of the questions. She gives you a condition and you have to say what happens. So which of the following enzymes would be upregulated when you can't break down glycogen? Under Le Forest disease. Try to do it without looking at the answer.
I have like a side question.
Go ahead.
Okay, do you think it's important to go and like memorize the names of these diseases and like what enzymes are affected, or do you think she would like say, oh, in Le Forest disease, like this specific enzyme does not work, or like this enzyme is deficient? Like then what happens? Does that make sense?
I would not. I think most of the time you could ask, even if she talks about it and she doesn't give you context, you can just ask her about it.
Okay, thank you.
Yep, so I wouldn't worry too much about that. I would focus on more of the big picture stuff. Okay, so enzymes that would be upregulated or downregulated, anything? What would be downregulated in Le Forest disease? How about that? That's an easy one.
Would it be glycolysis because you can't get there, or?
Right. So you can't make glucose, so glycolysis won't be running. So glycolysis would be one of the ones that are downregulated. What about glycogen phosphorylase? Would glycogen phosphorylase be active in Le Forest disease?
I want to say no.
Nope, it won't, because Lefort's disease causes insoluble glycogen. So phosphorylase won't be able to go in there and break it down. TCA would not be active. Complex 1 won't be running. Because you don't have the glucose that you get from glycogen, a lot of the stuff will be downregulated.
Okay, I have another potentially dumb question. So,
okay,
how do we know that stuff after glycolysis won't like remain normal because we'll get sources of acetyl-CoA from other places like beta oxidation? Or is that just like way overthinking the question?
I think that's way overthinking the question, yeah.
So if she like asks us something in the form of a multiple choice question like that, we should just stick with like exactly what the question's asking and not kind of tangents.
So the thing with these questions is it creates the cycle. So you're like, oh, Lefort's disease, you don't have enough glucose, glycolysis would be downregulated. You should stop right there. You shouldn't talk about what happens when now glycolysis is downregulated, because that would be a different kind of question. So just stick to around like the disease. Where is Lefort's disease and the things around it that it affects. You're talking about acetyl-CoA with. Go ahead.
So like this stuff, even though like we might be able to get this other like acetyl-CoA from other sources, we should say that like the enzymes that would all be downregulated because of the lack of glucose that would be going through glycolysis, right?
Yes.
Okay.
Okay, so that would be Lefort's disease. So she might ask a multiple choice on that.
Wait, another question on that one. Sorry. For the enzyme at C, I don't know how to say that.
Glutamate.
Where is that? Like, in what pathway?
That's in cycle between at the astrocytes and the neuron right here. You also talk about it briefly in the urea cycle. Glutaminase helps convert glutamine into glutamate into the neuron, and then glutamate into glutamate into the astrocyte.
Okay, perfect.
So it helps exchange between these two. Okay. So Lefort's disease. What is question 3 is talking about a PFK deficiency. So she could give you which of the following options would explain why an increased PFK activity results in decreased glycogen. Okay. So why would an increased PFK activity result in decreased glycogen? The reason is because PFK would make more fructose 1,6-bisphosphate will make less glucose 6-phosphate. There's other enzymes in the middle here that I'm not going to include. So we'll have less glucose 6-phosphate. So we'll have less glucose 6-phosphate being stored as glycogen. Okay? So that's why increased PFK activity would result in decreased glycogen. Okay. Question 4 is talking about the glyoxylate cycle. Do you guys... Actually, this is really low yield. I don't think she'll ask about this. But glyoxylate cycle is in plants. They take... What do they take? They take isocitrate, I thought. They take isocitrate, use an enzyme called isocitrate lyase. You make glyoxylate, and then glyoxylate and acetyl-CoA run through an enzyme, and then you I forgot. Does she have the enzyme name? I don't think I have it here. But you eventually make malate and you bypass a bunch of the electron carriers. I don't think it's that important to know. I don't think you'll be asked about glyoxylate at all. But it's in plants. And it's a way to bypass the bottom half of ETC. Okay. 5. What is this? For each of the following, draw the substrate products for the following. Okay. Glutamate dehydrogenase. Know stuff that glutamate dehydrogenase catalyzes. So which of the following, she could ask which of the following enzymes could catalyzes the conversion between alpha-ketoglutarate and an amino acid to glutamate and alpha-keto. So we're converting alpha-Kg plus amino acid to glutamate. Plus keto acid. So if she gave you this reaction and so she gives you this reaction, what are and your options are a transaminase, glutamine synthase, alpha ketoglutarate dehydrogenase. Yeah, out of those three, which one would you guys pick? Or glutamate dehydrogenase, by the way. Go ahead, Angela.
Is that the transaminase?
Yep. So make sure you know the difference between the transaminase and the glutamate dehydrogenase. Glutamate dehydrogenase does the same thing, except it uses NAD and NADPH instead of the amino acid and alpha-keto acid.
I have a question about transaminase reactions.
Go for it.
Okay, so you know how like most enzymes like very specifically use one thing or like two things and go to two other specific things? Is the transaminase reaction is it one enzyme that does like any amino acid to an alpha keto acid, or is it like different transaminase enzymes? I don't know if that makes any sense.
I think they might have general names, but I think they're just all called transaminase. I don't know though.
But it is like different enzymes, so it's not one enzyme that does like every single.
No, it's not. It's not, because you would have to fit different like substrates in there, so it would have to be a different enzyme.
That just confused me for a while, but okay, now that it's a class of enzymes then.
Yep. Transaminase are just a class of enzymes that transfer that transfer the amino group.
Thank you.
Okay. How does
that? So I just want to make sure I understand that you say transaminase and like it's like at the same level as like a kinase, right? Like a kinase does one thing and transaminase does something. It's like at the same level.
Yep. So kinase is like a class of enzymes that add phosphate. Transaminase is a class of enzymes that transfers an amino group.
Okay, perfect. Thank you.
But we don't need to know the different ones. Like obviously we need to know the kinases.
No, I don't think you do.
Okay,
I don't. So I don't think you guys are expected to. Okay, so 5B. She could ask, how does fructose 2,6-bisphosphate regulate PFK1 and fructose 1,6-bisphosphatase? So which one does it inhibit and which one does it activate? This one.
It inhibits. So the F2,6-BP inhibits fructose 1,6-bisphosphate and activates the PFK1.
Yep, that's correct. So inhibits the fructose 1,6-bisphosphate and activates PFK1. So remember, because it's made up by PFK2, you make fructose 2,6-bisphosphate and you activate PFK1. Which of the following enzymes help make NADPH in the pentose phosphate pathway? There's two of them. Do you guys know the two?
Is one glucose 6-phosphate dehydrogenase?
Yes, because it's right here, Maddie.
Sorry, that was from my brain. But
no. You're good. Okay, so that is one. What's the other one?
Phosphogluconate dehydrogenase?
Yep, the other one is the one that has the dehydrogenase in it. So remember, you have the glucose 6-phosphate dehydrogenase and then the 6-phosphogluconate dehydrogenase. This one does not make NADPH. So those are the two steps that make NADPH. Okay, so that was 5, 6.
Can you say the name of that second one more time?
Yep, 6 phosphogluconate dehydrogenase. So those are the two that make NADPH.
Thank you.
Mm-hmm. Okay. If cells are fed 13C glucose, explain how the following metabolites would be labeled. Okay, so you're giving cells 13C glucose labeled at 1, 2. How would these metabolites be labeled? Okay, how would glucose 6-phosphate be labeled? How many labeled carbons would it have?
2.
Yep, so glucose would be converted to G6P. So all of the 2 carbons that are labeled would be labeled in G6P. What about fructose 1,6-bisphosphate? How many carbons would that have labeled?
2.
Yep, so that one would have 2 as well. Okay, what about 2 phosphoglycerate? How many would that one have labeled?
Would half of the 2PG have 1 and 2 labeled, or like 2 of the C's labeled, and then the other half would have none?
Yep, that's right. And that would be the same with the 3BG. That's good.
Do you mind explaining that actually?
Yep, let's go to glycolysis. Let's go to the step where it splits. Okay, so remember, up until this point, we haven't done any carboxylation reactions in glycolysis, so we're at fructose 1,6-bisphosphate. So our carbon 1 and carbon 2 are labeled. So when we split by aldolase will have, so they switch the like numbering of the carbons, but DAB would have 2 carbons labeled and GAB would have 0 carbons labeled, okay? Based on the way we split, so we split right here. So DAB would get 2 of the labeled ones and GAB would get none. But because remember there's an enzyme that converts between GAP and DAP, when we're running glycolysis, the DAP is converted into GAP. So the DAP that had 2 carbons labeled will become a GAP that has 2 carbons labeled. So what we end up with is a GAP that has 0 carbon labeled, and then another GAP, so this one comes from DAP, that has 2 carbons labeled. Okay? That's why you get 50% of them labeled with 2 and the other 50 are not labeled.
Okay, that makes sense.
Makes sense? Okay.
Yeah, thank you.
Mm-hmm. Okay, question 7. So this is on the pentose phosphate pathway. So in order for a hepatocyte to synthesize fat, what other pathway need to be upregulated? What do we need to synthesize fats? What are
I would say the oxidative phase of the pentose phosphate pathway would need to be regulated, and then the beta oxidation pathway would need to be downregulated using ACC2.
Yep, that is right. Down regulates CPT1. Okay, so that would be good for 7A. A lot of this is talking about oxidative phase and non-oxidative phase in the pentose phosphate pathway. 7B, reactive oxygen species favor the production of what pathway in the cytosol? So which phase of the oxidative phase would be favored when we have reactive oxygen species? Remember, reactive oxygen species, those are dealt with by glutathione. So to help with that, you need to be making NADPH, because glutathione helps oxidize reactive oxygen. Is it oxidize or reduce? Oh, let's go. Forgot what it does exactly. Yeah. So glutathione helps take reactive oxygen species. So for example, hydrogen peroxide, oxidize them into water, and in that process, it gets, so that would be, it gets oxidized. So to put it back into the reduced form, no, I'm sorry. It helps reduce hydrogen peroxide into water. That's why I was confused. So we reduce hydrogen peroxide into water, and then glutathione gets oxidized. So to help put it back in the reduced form, we need to make NADPH, and this comes from the pentose phosphate pathway. Okay? So that would need to be upregulated when you have reactive oxygen species. If you have a proliferating cell that needs large, a proliferating cell, so like a cell dividing a bunch, needs large amounts of what molecule from the pentose phosphate pathway? Remember, you need to run. Oh, go ahead, Kaylee.
NADPH.
No. Remember, NADPH is used for fatty acid synthesis and reactive oxygen species. So remember the non-oxidative phase. One of the things it makes is ribulose 5-phosphate, which then can be converted into ribose 5-phosphate, and then that can be used for nucleic acids and helps in a cell that is dividing, because a cell that is dividing needs the ribose 5-phosphate for their nitrogen base for their nucleic acids. So we need to upregulate the non-oxidative phase to help make the ribose 5-phosphate. Does that make sense?
Yeah, thank you.
Mm-hmm. Yeah, it gets confusing with the oxidative and non-oxidative, but usually oxidative will be to synthesize fats and to deal with reactive oxygen species. Okay, so we have tumor cells that proliferate a lot. So they have upregulated. What do they upregulate for energy, and what do they upregulate to be able to divide really fast? So they usually upregulate glycolysis. So tumor cells do a lot of glycolysis, so they'll upregulate glycolysis for energy, and they'll upregulate the pentose phosphate pathway non-oxidative phase. They make ribose 5-phosphate to be able to divide really fast. So tumor cells and cells that proliferate a lot will need to run the non-oxidative phase. Okay. Okay, so the next question is about ETC and the Q cycle. How do you guys feel about the Q cycle? Or do you want me to explain that?
Honestly, if you could just kind of talk about the Q cycle, that would be much appreciated.
Yep, I can do that. Because the part A is talking about the Q cycle. Let's see. Okay, so we have reduced coenzyme Q that comes from complex 1 and complex 2. So reduced coenzyme Q goes into complex 3, has 2 electrons, so it gives 1 electron to an iron sulfur subunit, and then it gives the iron sulfur subunit gives the electron to a heme group on another protein called cytochrome C1, and then cytochrome C1 gives 1 electron to cytochrome C. Okay, so the whole point of the Q cycle is cytochrome C can only accept 1 electron at a time, but we have 2 electrons. Okay, so we need to kind of run it twice to be able to give our 2 electrons to cytochrome C. So if we go back, so remember we said reduced coenzyme Q gives one electron to the iron sulfur. The other electron would be passed to a group heme called heme Bl, and then it's passed to another group heme BH. So this is the high potential heme, this is the low potential heme. The high potential heme would then give the electrons to an oxidized coenzyme Q. So this comes from previous cycles. So it's already oxidized, it gave both of its electrons away. We give our second electron to that, and then we create this superoxide. Okay? Then we have another, so this is a different coenzyme Q. You have a lot of these. You have a lot of these QH2s. So the one we just oxidized right here, that one can receive electrons and be reduced again. Or you can have a brand new ones that come in. So we have another coenzyme Q, reduced one, comes in, goes through the same cycle, gives 1 electron to iron sulfur, give 1 electron to cytochrome C1, and then cytochrome C1 gives 1 electron to cytochrome C. So far we successfully transferred 1 electron to cytochrome C here and 1 electron to cytochrome C here. Okay, so this coenzyme Q gives its second electrons to the low potential heme and then gives it to the high potential heme. And then the superoxide that we made in the previous cycle receives the last electron from the second reduced coenzyme Q. And then we get a full reduced coenzyme Q again that can start a new cycle. Okay, so we use 2 reduced coenzyme Qs. So each would have 2 electrons, so that's 4 electrons total. 2 of the electrons went to cytochrome C, and 2 of the electrons went to regenerate a second coenzyme Q. So in total, we transferred 2 electrons. We used 2 reduced coenzyme Qs, but we also made 1 reduced coenzyme Q. So that's why when you show it in the pathway, 2 electrons go from coenzyme Q straight into cytochrome C. Okay? Does that make sense?
So are we pumping 4 electrons? 4 hydrogen, so 2 for each electron, is that right?
Yep. So whenever we pass the electrons, so the first round when we pass the iron sulfur and low potential heme and high potential heme, we pump 2 protons. And then the second time we do it as well, we pump 2 more protons as well. So we pump 4 protons, transfer 2 electrons, and then we regenerate one of our reduced coenzyme Qs.
Okay, and then just kind of like side opinion question again. Is it something that we should kind of like be able to recite like you just did, or is this something that's more so like understand the reason that we do it kind of thing? Because I don't know if that makes sense.
I could see her asking an extended response on that, but if you don't have time, I would know the general concept of it. That you can say that the whole goal of the coenzyme Q is we have 2 electrons, but cytochrome C can only accept 1 at a time, so we need to run the cycle twice to be able to give 2 electrons to cytochrome C.
But you definitely think it's fair that she could ask like an open-ended question.
Oh yeah, I think so. The Q cycle, pyruvate dehydrogenase complex for sure. I don't want to say for sure, but Q cycle pyruvate dehydrogenase complex. And then she could do like, I don't know, the other ones too. She will definitely give you stuff where you have to draw some substrates. So that might give you like 4 enzymes and you have to draw their substrates and products. So another question like that. I'm not sure, but the Q cycle definitely is one of the really good ones to ask on extended response.
Thank you.
Okay, so AA. So let's talk about, so this is talking about uncoupling proteins. So how does uncoupling proteins affect the electron transport chain in ATP synthase? Does anyone know?
Doesn't it mess with the proton gradient?
Right. So what would that do to the electron transport chain?
Well, if you don't have a proton gradient, then you can't run ATP synthase.
So ATP synthase would not run. Would the electron transport chain run faster or slower?
Faster?
Right, it would run faster because the whole point of the electron transport chain is to make this proton gradient. So when you dissipate that proton gradient with the uncoupling proteins, the electron transport chain will try to work harder to get the proton gradient. Okay, and then ATP synthase, of course, because we don't have a proton gradient, would be inhibited. Okay, so we also just talked about IF1 at the beginning. IF1 inhibits ATP synthase, and I think she has the explanation for that over here. And then Y's inhibition of ETC results in downregulation of pentose phosphate pathway. So I think she has that explained. When inhibited complex 3, ATP production decreases and flux through glycolysis increases. So there will be less glucose 6-phosphate available to produce glucose 6-phosphate dehydrogenase. So production of NADPH will go down. So basically she's saying because ETC is inhibited, glycolysis is running faster. Because glycolysis is running faster, there is less glucose 6-phosphate. So less glucose phosphate is going into the pentose phosphate pathway. So that's why inhibition of ETC leads to downregulation of the pentose phosphate pathway. Okay. 9. What is this?
I have a question about that, sorry.
Yeah, go ahead.
Associated glycolysis, like if you have lots of glycolysis, you would also have lots of pentose phosphate pathway, because if you're able to run glycolysis, then obviously you have like enough glucose in general to then be like, I guess I just thought that like when you're running glycolysis, you also have enough resources to run the pentose phosphate pathway.
Usually that is the case.
Yeah, is it just because we're inhibiting the electron transport chain, so we have to like focus only on making energy for the cell, but in like other cases that usually is true?
Yes, usually that is true, but based on her explanation, I think that's what she was going for. So, but your thought process is correct. Usually when you have a lot of glucose, you shunt some of it into pentose phosphate pathway.
Thank you.
Mm-hmm. Okay, so question 9. You are studying oxidative phosphorylation. You treat the cells with DNP, which acts as uncoupler, and then you measure lactate production. So 9A is talking about uncouplers. So uncouplers upregulate what pathways? Does anyone know? So uncouplers, go ahead, Allie, go ahead.
I was going to say, didn't we just say that an uncoupler would upregulate glycolysis?
Yep. So uncouplers will, well, the first thing they will upregulate is ETC, right? They'll upregulate ETC, and then we'll downregulate ATP synthase, so we're making less ATP, so glycolysis would run faster.
Sorry, with that being said, then we would also increase TCA to keep making the reduced electron carriers for the electron transport chain, right?
Yes. Okay, yep.
So then, I'm sorry, I have multiple questions.
No, it's good. It's good.
When it's uncoupled, then since we're upregulating TCA as well to be able to make those reduced electron carriers, we don't have to use fermentation, right? Because the electron transport chain and TCA are still both working, right? I just want to make sure.
That's a really good question, huh? Let me think. Well, lactate production increases. So I guess it is running fermentation because this is an uncoupler to T4 DNP. So we are increasing lactate production. So there is fermentation would definitely increase. Does she talk about? So the way she explained it, she said if there are not enough NAD and FAD acceptors for TCA to match the speed of glycolysis, pyruvate will get reduced into lactate. So most of the pyruvate will go into lactate because we're not glycolysis is moving faster than we're generating NAD and FAD for TCA to use. So that's why you get the increase in fermentation. And I would say TCA stays the same for the most part, I guess. I know I said upregulated the most, but now seeing this graph, I would say TCA stays the same or decreases actually.
Wait, can you explain why TCA would decrease if the electron transport chain is increasing?
The electron transport chain is increasing, but
because like does it get a majority of its electron carriers? Okay, wait, sorry, this is just another thought. I could be totally wrong, but the electron transport chain is obviously just using the reduced electron carriers. If glycolysis is like running fast enough, could it provide like a majority of the reduced electron carriers for the electron transport chain? So then we don't really have to worry about what TCA is doing.
Yeah, and the cell also has enough. So you're talking about the NADH that glycolysis makes, right?
Yeah, because if the whole logic behind this question is that glycolysis is running so fast that there's not enough NAD plus available, then obviously the electron transport chain isn't keeping up with reoxidizing those things fast enough. So
yeah,
my thought is maybe all the reduced carriers are coming from glycolysis instead of TCA.
Yeah,
because glycolysis is running so fast.
Yeah, I guess that makes sense. And that would explain why fermentation would increase. Because fermentation also regenerates NAD for glycolysis to use, so glycolysis can just use its own electron carriers.
So basically the idea is that glycolysis is running so fast that even though we're upregulating the electron transport chain with this uncoupler, it's still not enough to reoxidize all the stuff that we're using in glycolysis because it's running so fast. So then on top of using the electron transport chain, we also have to use lactate.
Yes.
Fermentation.
Yes,
okay.
I would say that's the best explanation for that. Okay. Question 2. HIF1 is activated under what conditions? So 9B is talking about HIF1, so she could ask you about what HIF1 is activated under. So you guys know what conditions HIF1 is activated under? It's called hypoxia inducible factor, if that helps.
So hypoxia.
Yep. So it's one of the things that is activated under is hypoxia. It can also be activated by low energy levels as well. So low ATP, high AMP, that would activate it as well. What happens to oxygen consumption when an uncoupler is added?
Can you repeat that? My bad.
Yeah. So hypoxia inducible factor activated under hypoxic conditions and low energy conditions. So high AMP, low ATP. Okay, 9C, what happens to oxygen consumption when an uncoupler is added? So we already said that ETC runs faster, so oxygen consumption would increase. 9D, not sure what it says. So 9D is considering. So 9D was asking about if you add D and P and then you add compound C. You can explain what happens with the decrease in lactate addition. So 2,4-DNP is added, you increase lactate production, and then we add compound C, and then lactate production decreases. So I don't see her asking anything outside from that. She in case you're given something like that, it would probably be a graph, and you would have to interpret it the same way for 9D. Okay.
I want a quick question on that one. Sorry.
Yeah, go ahead.
Before we were talking about how I know Maddie asked, because she assumed that every time glycolysis increases, so does the PPP, which I also made that assumption.
Mm-hmm.
But I was wondering if in this case, since NAD pH is needed for a lot of things that happen at high energy, if that would be a reason that PPP isn't running because we're in the low energy state, or is the PPP at all connected to higher low energy state when it's running, or?
That is a correct way to explain it. So if you put that down, that would be marked right. A lot of these questions have multiple answers. Jesus explained it because she said there's a lack of glucose 6-phosphate available to go into PPP.
So can I like make the assumption that the PPP is like related to a high energy state, and when it's in a low energy state, the PPP will always be decreased?
Yeah, yes. It's not good to like sometimes having like these shortcuts where if this happens right away can get you
like at least most of the time.
Most of the time, just don't apply it to everything because sometimes there's exceptions, of course.
Okay, I just want to make sure that wasn't like an unfair comparison.
No. It's definitely that would be a good explanation for that question.
Thank you.
Mm-hmm. Question 10. So this is talking about you're given a bunch of proteins and you have to explain what the normal function of the protein. You have to talk about the structure that it facilitates. So for ATP synthase, so what two subunits does ATP synthase have, and what are their functions respectively? Do you guys know? Because she could ask you a multiple choice on that.
F0 and F1.
Yep, F0 and F1. So F0 acts as a proton channel.
Proton channel, yes. And F1 is the ATP synthesis.
Mm-hmm. So remember, proton channel, protons go in, allow ATP synthase to spin, and then that causes conformational changes, and then ATP synthase allows us to make more ATP.
Yeah, it will push the ATP from the surface of the F1.
Mm-hmm. Correct. I think she also has, talking about the shape, it's shaped like a turbine. Each hydrogen is translocated, and it turns it. Okay, so transketolase. She has a cleft arginine, histidine, serine, aspartic acid, glutamic acid lining the active site, and then these interact with the phosphate group. I think the important part to take away from 10b is it uses TPP as a cofactor. Glycogenin. What is the role of glycogenin in glycogen synthesis? You guys know?
That's the big guy that starts the whole process,
yep,
right?
That's the big guy that starts the whole thing, pretty much. That's what glycogenin does. Cytochrome C, we just talked about this one. What two subunits does it allow exchanging of in ETC?
I'm pretty sure complex 3 and 4.
Yep, complex 3 and 4. Complex, remember, we just talked about in the Q cycle, takes electrons from cytochrome C1 and then goes into complex 4. And then gives the electrons to water, and then water is reduced. Or not water, gives the electrons to oxygen, oxygen is reduced to water. Okay, question 11. What effect, if any, would acidification of the cytosol have on this? I think for this question, she might give you one of the ones where which of the following would happen. One of the scenarios one. So she would tell you that cytosol is acidified, and you would have to say what happens to certain pathways. So that's what she could ask from this question. So PFK, acidification of the cytosol. Remember, protons inhibit PFK, so glycolysis would be downregulated. So for 11D, flux through glycolysis, that would be downregulated, and because PFK is downregulated as well, or PFK is inhibited. Oxygen consumption. So she said that's her explanation for this one. I think I may have, people who came in asked me for help, I gave you guys a different explanation for that one, but go off by her explanation. So she said the low pH in the intermembrane space makes pumping protons harder, so then you have less oxygen consumption. ATP production, the low pH in the inner membrane space, said increases the proton motor force, and then that increases ATP production. And then flux through TCA, she said that would have lower flux because less oxygen is consumed, so we don't need to make as much electron carriers. We have fewer NAD pluses to go into TCA, so flux through TCA is decreased. Okay, 12. So this.
Wait, can I ask a question about the last one?
Mm-hmm.
Is there like I just want to make sure that this is a logical, I guess, reasoning. Like physiologically, do we want to inhibit PFK from running when we're already in an acidic state so we don't get like too acidic? Because if we keep running glycolysis and like eventually we're going to go to lactate production, which is just going to make things like more acidic. So is this kind of like a feedback mechanism to prevent the cell from becoming like acidic so that it doesn't die?
Yeah, usually this will happen in a muscle cell that's contracting really fast. So when a muscle cell is contracting really fast, the cytosol will be really acidic from all the lactate that's being produced. When the muscle is contracting really fast, you want a way to inhibit it and tell the muscle to stop contracting because we have a really acidified cytosol. So then those protons go back and inhibit PFK. So the muscle doesn't just keep contracting forever without stopping. So yes, it is like a feedback inhibition mechanism.
Thank you.
Mm-hmm.
Hey, Manuel, I was gonna head out, but
okay.
I wanted to ask you. You said you were gonna post these review sessions.
Yes, I will. It might take like 5 hours to upload because it's getting really long. And I will post the rest of this because we'll only get through problem set 2, and then I'll post the other ones.
Okay, will it be like a view only type of this?
Yeah, I will see if I can do that. Even if it's edit type, if I share it, hopefully no one goes in and deletes the stuff.
Right.
But yeah, it would be like view only where you could see this.
Okay, thank you. Appreciate it.
Yep, no problem. Good luck on your test.
Needed. Thank you.
Okay, 12. So we have 12 to 20. So I'll go over those and then we'll call it a day. 12. It's talking about oxidation reduction. So this is, you want to be able to use the Nernst equation for this. So Nernst equation is negative delta G is equal to negative NFE. So N is the number of electrons, F is Faraday's constant, and E, actually, I don't like E. We'll do delta E. So this is the change in electric potential. Okay, so, and then you calculate delta E from the reduction potentials given. Remember the one that is reduced, you leave it the same, and the one that's oxidized, you flip it and then you add both. Or you can subtract a negative like they did here. You can do it differently to get the delta E. And then you plug in the delta E into the Nernst equation, and then you get the delta G. Make sure you're also able to calculate it the other way as well. So if you're given the delta G, you'll be able to calculate delta E. And then make sure all your units match. So usually delta E will be given in volts. N is just number of electrons. And then Faraday's constant will be given as, it's 96,000, and it's given in joules per volt. Joules per volt is the same as coulombs. Volts would cancel with volts, and then it will end up with joules as our delta G. Okay. So yeah, this is all about the Nernst equation. For 13, for each of the following, indicate whether the addition of an uncoupler compound such as ENP would increase, decrease, or have no change in the flux through the pathway. Explain your reasoning. Okay, so this is again with uncouplers. So let's briefly go over them. So if you add an uncoupler, ETC would increase, ATP synthase would not run. So you will have low ATP levels. This would upregulate glycolysis. See, Maddie, I don't she kind of like contradicts herself here because she said TCA increases here. But for the other
so
one,
maybe it's like some sort of combination between glycolysis running really fast and TCA still running and doing its thing.
yeah. So I'm not sure because she says.
I think.
Go ahead, Kaylee. Go ahead.
Oh no, I was just gonna say I remember her talking about that one and saying the word overflow. I don't know if that's a helpful jogging memory thing.
Overflow. So increase because need ATP stimulates PFK.
Do you mean, Kaylee, do you mean she was saying like fermentation like deals with the overflow of reduced electron carriers? Is that what you're saying?
Yeah, I'm pretty darn sure, but I don't have it in front of me.
Okay, no, that makes sense to me. If that's what you're trying to say, then that makes sense.
Yeah. Thank you.
Okay, I'll ask her about it tomorrow in the review session because I'll also be there. So I'll bring that up to her because I'm confused now about it. What happens to TCA? Okay, so we said glycolysis increases. So DNP will make so it's an uncoupler. ETC is upregulated. ATP synthase is inhibited. We're in a low energy state, so we'll break down fatty acids. So beta oxidation would increase. Glycogen breakdown would increase because we're in a low energy state. Gluconeogenesis. She said this one would increase in the liver. So this is why it's important to know what tissues we're on. For all of the other organs will increase glycolysis to make energy, but the liver will focus on gluconeogenesis to give the other organs glucose. Okay, so that's why it's important to keep track of what tissue we're in, especially with glucose and gluconeogenesis. And then the pentose phosphate pathway would decrease. Like Ali said before, this is run when we need to make NADPH to synthesize stuff. So if we're in a low energy state, DNP is inhibiting ATP synthase, we probably don't want to run the pentose phosphate pathway. Okay, so that's for 13. 14, so we are adding oligomycin. So this blocks the proton channel F0. And we have to explain why lactate concentration builds up in rats when we add this.
Can I ask you a question about the previous one? I'm sorry.
No, go ahead, Maddie.
Okay. Okay, so originally I put in my problem set that gluconeogenesis would decrease because the body's in a low energy state and it requires 6 ATP to be able to produce more glucose, which would then only make 2 ATP by like breaking it down in glycolysis. So I said that you wouldn't increase gluconeogenesis. So I guess it just doesn't make sense that it's going to cost more ATP than it would to use it in glycolysis. Yet we're still increasing it. Like that just doesn't make sense to me.
Did she mark it wrong?
No.
Okay. I guess if you explained it well and you have good reasoning, that could make sense.
Yeah, I think I talked to you about this one, and that made a lot of sense to me. So that's why I guess I'm confused.
Yeah. So I would have originally said gluconeogenesis would decrease, but I think she's talking about we add DNP here, we add DNP here. Those ones have low ATP.
Oh, so you're saying like it's tissue specific? So like the brain and muscle have low ATP, but the rest of the body's in like a normal state. So the liver's willing to kind of do that to help the brain and the muscle out. Yeah,
pretty much. I think that's what she was going for here. If you're ever confused, just ask her what tissue we're adding this stuff in.
Okay, so if this was talking about like the entire body having an upcourse, so everything's in a low energy state, it would decrease. Gluconeogenesis would decrease because everything's in a low energy state. You wouldn't want to waste energy to do that. Is that right?
I don't want to give you a wrong answer. Originally, I would have said yes, but I'll ask her tomorrow about it just to make sure.
Thank you.
I just don't want to give you something wrong, but I'll ask her about that one for sure. But I think this is what she was going for. I don't know what happens if you add DNP in the liver. I'll ask her about that tomorrow. Okay, so we add antibiotic, oligomycin. We block this proton channel, so we block F0. We have to explain why lactate concentration builds up in rats that have been treated with oligomycin. So this is talking about why glycolysis would be upregulated upon the addition of an antibiotic that inhibits F0. So this is really similar to all the stuff we've been doing the past few questions. When you block the proton channel, you won't be able to pump protons through it, so ATP F1 won't be able to make ATP, so that will cause a low energy state. A low energy state would cause glycolysis to be upregulated. So she might give you like options, and you would have to select which one best explains why glycolysis is upregulated when you add an antibiotic that blocks the F0. Okay. 15. Oh, I made a multiple choice for you guys.
Well,
yeah, go for it.
sorry, I have a question for the last one.
Mm-hmm.
It takes me a second to wrap my head around my question. So the whole purpose of like taking pyruvate and like making it into lactate is to make more NAD plus, right, for glycolysis?
That's not the whole point of it.
Or like the reason that like you don't just have like a buildup of pyruvate?
Yeah.
Okay, so sorry, I just want to make sure that was correct before I go into my question. So in this case, if you were like running like only F0 is like blocked, then would you still be running like the TCA cycle and then the electron transport chain, like making NAD plus like from the electron transport chain? And then in that case, why would you make lactate? Because you already have NAD plus circulating from the electron transport chain.
So you're asking why the electron transport chain is not running?
Yeah, because F0 is in ATP.
Yeah, but they're usually coupled together. So if you're not pumping protons, right? What does H0 do? It pumps it's a proton channel, right? Let me pull it up.
Yeah.
So it will act. Let's see. Because they're coupled together, it will eventually affect ETC as well. It pumps protons from the intermembrane space into the matrix, right? ETC takes the protons from the intermembrane space, takes protons from the matrix and puts them into the intermembrane space. So if the intermembrane space becomes really acidic because this is not working, the electron transport chain will think that it accomplished its job because all the protons are already here. So the electron transport chain would not run.
Okay, that makes sense. Thank you.
And then when the electron transport chain is not running, we're not making NAD and FAD for TCA to run, so TCA won't run either.
Yeah, that makes way more sense.
Okay, sounds good. Okay, let's see. So this is about ETC. So she's talking about changes in NAD, NADH, and ATP and ATP that occur from switching to anaerobic to aerobic. Okay, so I actually made a multiple choice for you guys. Which of the following explains what happens to ATP-ADP ratio and the NAD-NAD ratio when switching from anaerobic to aerobic respiration? So when we switch from anaerobic to aerobic, what happens to the NADH and NAD ratio? Would that increase or decrease?
This is just a thought. I could be wrong though. I feel like it would. My guess would be that it would increase because we're running. We'd be running the TCA cycle, which would be making more NADH. But I also feel conflicted because we'd also be running the electron transport chain, which would be getting rid of that.
No, so the second thought of is just you like overthinking this. These are electron carriers. you make NADH to put it into ETC. So whenever you're running aerobic respiration, you'll have more NADH. So NADH to NAD ratio would increase. Okay, so that was the hard one. What about ATP to ADP ratio? Would that increase or decrease? The ATP to ADP ratio. We're running ETC.
That would also increase.
Yep, so we're running the ETC. So more ATP is being produced. So it would be this one.
Okay, so this is a dumb question, but how do you know when to like stop thinking about things? Because I definitely would have sat there and like had a 20-minute debate with myself on the exam about what I think about the electron transport chain or not. So I'm just wondering, like, how do you know to not think about the electron transport chain?
Usually, I don't know, because I overthink a lot of questions all the time on the test, and that's mainly why I get a lot of questions wrong, so I'm not the best to give you advice on this. Usually, you want to look at it from one step. Most of the questions I missed when I took this class was because I was like overthinking it. So I think about it really simply. NADH is an electron carrier. It will be upregulated during aerobic respiration because it helps give electrons to the electron transport chain. So we're looking at a ratio from point A to point B. Maybe overall NAD and NADH's concentration would increase, so both of them would increase. But from point A to point B, there will be more NADH.
Okay, if is that like a fair question that I could ask on an exam? Like I could ask her and be like, hey, do you want me to just think about TCA or also think about ETC, or do you think that's like kind of overstepping what you could ask on an exam?
No, I think you could just you can ask her about it and get more clarification. But the best way to do it is like one step from where you're at. So we're switching from anaerobic to aerobic. What is going to happen? We're going to make NADH through TCA, and that's going to go into the electron transport chain. Okay? After that, you're overthinking, I feel like. And that happens with all the pathways as well.
Yeah, I think that's most of the time my major issue. So
yeah, yep, no problem. I made the same like mistake a bunch last year as well. Okay, question 16. So this is talking about know the structure and function of LDL, HDL, and VLDLs. So let me just go over them real quick. Oh, no, I added a picture that has these. Whatever. So chylomicrons, they originate in the small intestine. They're made of tags. They take dietary tags and cholesterol and give them to the liver. VLDLs originate in the liver. These ones, and they're also made of tags. They distribute tags from the liver to other tissues. So chylomicrons bring stuff from the intestines to the liver. VLDL takes tags away from the liver and gives them to target tissues. LDLs originate in the liver and they distribute cholesterol to other tissues. And then HDLs, they're also made of cholesterol. HDLs are also originate in the liver, but they return cholesterol back to the liver. Okay? So if we have the liver in tissue, LDLs take cholesterol away, and then HDL return it. Okay, next question. Question 17. Yep, no problem. Question 17. Imagine you are the fifth carbon in oloic acid and part of a tag. Outline your path from the lumen of the small intestine to the final oxidation in the matrix. So you could ask, describe, and draw in full detail the outline of how fatty acids get from the intestine to the mitochondrial matrix. So I think I'll just go over some pictures for that one because it will be easy. Is it? I can't remember if it's this one or the other one. It might be the beta oxidation. Yeah, it is the beta oxidation. So let's see this. Let's go back to the first one. So uptake of fatty acids in the intestines. So you have your fatty acids. You use what are they called? Bile acids to emulsify them, and you put them into a missile. The missile is then absorbed into the intestines. The intestines take the missile, you break it down into individual fatty acids, and then you make it into a chylomicron. You send the chylomicron into the blood or lymphatic systems. Usually it will go to the liver. After it goes to the liver, it can get repackaged, or in our case, we're talking about it going into the mitochondrial matrix. You can also, no, this is not. So the first thing that we'll do, we'll run through a lipoprotein lipase. Lipoprotein lipase hydrolyzes the fatty acids, the chylomicrons, and then we'll get free fatty acids and glycerol. The free fatty acids then diffuse into the cytoplasm. After they diffuse, you run a reaction called acyl-CoA synthase. You take the fatty acid and coenzyme A, and you make fatty acyl-CoA. Once you make fatty acyl-CoA in the cytosol eventually enters the matrix through the carnitine shuttle. And I explained this in more detail in my video. But once it enters the matrix, we're able to break it down into beta oxidation, and then that can go into TCA. So I think that's really similar to how she talks about it. So she said absorbed in the intestinal cell, and then interact with this to make a more soluble, reattach to glycerol to form tag, patch to become a chylomicron, released into the lymphatic, goes to the liver. She also says you can go into the muscle and adipose tissue. You run through a lipoprotein lipase. You produce fatty acids and glycerol, taken up into tissue cells. And then you pretty much transfer, go through the carnitine shuttle, and then go inside the matrix, and then you go through beta oxidation, and then enter TCA as carbon-1 in citrate. Yeah, that's pretty much everything.
I have a quick question about the slide that you were on before.
This one?
Yeah, originally, no, wait, sorry. The one with the carnitine shuttle. I guess you were on a lot of slides, so I guess I needed to be more specific.
No.
One.
Okay.
Okay, so the step that goes between the outer membrane, that goes from the cytosol into the intermembrane space, that's CPT1,
Mm-hmm.
and then that next step is the carnitine transporter, just like some protein in the membrane.
It's a different transport. It's a protein in the membrane. Yep.
Okay, and then CPT2 is once you're in the matrix, it then turns that back.
Yep.
Okay, for some reason I was under the impression that CPT1 goes across the outer mitochondrial membrane, and then CPT2 goes across the inner, but obviously that's wrong, so I just wanted to make sure that I know it.
They catalyze the same reaction, they just go the other way, and they're in different locations.
Okay, so the carnitine transporter is like a protein in itself that's embedded in the inner mitochondrial membrane.
Yes, it's a different transport.
Thank you.
Mm-hmm. Okay, so that's pretty much it. So she might ask you to describe. So this could be one of the extended response. I don't see her how she could else ask this as a multiple choice question. So you would have to describe how fatty acids get from the intestines into the mitochondrial matrix. What two enzymes are required to degrade fatty acids with two cis double bonds that are at the 9 and 10 position, or 9 and 12 position? So for this, you have, I don't think you'll ever get asked on this, but you have two cis double bonds. So remember, beta oxidation wants to occur when you don't have any double bonds at all. So we want to convert, or no. It doesn't want cis double bonds. It's okay with trans double bonds. Because remember, the first step in a beta oxidation is converting our carbon chain into a trans double bond. So if we have a trans double bond originally there, we can kind of skip the first step. Let me explain this better. If we look at this. So the first step here, we have the fatty acyl CoA. First step is we make a trans double bond. So if the fatty acid is already in trans double bond, it kind of saves us a step. So that's what happens if you're in a trans double bond. If you have cis, you have to run extra enzymes. So there's an enzyme that helps you convert between cis and trans double bond. So do you guys know what this enzyme is called? That helps convert between cis and trans double bonds. Okay, it's called eno-CoA isomerase. Okay, so this one helps us convert between cis and double bonds. Okay, so that's if you have one cis double bond. The question is asking about the position 9 and the position 12. Okay, so when we switch the position 9 to a trans double bond, this will mess up the 12 double bond. This will put it in a different configuration. So we have to run through two different enzymes. They're called, I don't think you'll have to expect it to know these, but one of them is a reductase and then an isomerase. And then that puts the double bond in that trans configuration that enohydratase can use. So that's if you have two cis double bonds. You have to do these two extra reactions.
So we don't like the cis double bonds. We have to convert them to trans to be able to use them. And that's when we can skip, once we already have a trans double bond, that's when we can skip that first step.
Yes.
That makes a trans double bond.
Mm-hmm.
Okay, thank you.
And if you have two cis double bonds, you have to run an extra reaction because the configuration of carbon 12 gets messed up when you switch carbon 9. Okay. Question 19. So this is about techniques. So you want to compare the amount of HIF 1 alpha in cells in hypoxic conditions and normal conditions. Describe step by step two approaches you would take. So you just have to know that to compare protein levels, you can use Western blot or ELISA. And then you can also use mass spec as well to compare protein levels. It says describe step by step. So I don't have step by step for how a Western blot works, but I'm sure you guys can look that up online.
Do you think that with all the other material that's much more relevant to like biochem and metabolism that she'll ask us to write out like these detailed descriptions of how they work?
I don't think so. If I was studying for this, I would just know that a Western blot compares protein levels. I will feel really bad if you guys show up and you have to describe a step-by-step on our Western blot.
Because I know like some of them from like CellPhys, and I don't know if anyone else here is also taking CellPhys, but other than that, like she obviously didn't have really went over it other than this one question. So I just wanted to make sure you didn't think this was like super high yield or something.
I don't think it is. I wouldn't worry too much about this question. And then
I won't hold it against you though if it's on there.
Okay. Which technique allows comparison of protein levels? So for 19B, that would be the same thing. And then Western blot is faster and more straightforward than ELISA. So if you have to pick between the two, go with Western blot. Okay. Last question, 20. So we have a protein that's 750 kilodaltons. You do an SDS page. There's a single band at 250. Will the protein exhibit sedimentation corresponding in 250 or 750? So because it's only one band at 250, it was probably homo trimer. So homo just means they're the same subunit. They're the same size, and trimer just means there are 3 subunits. It's 750. You get one band at 250, so you just make the assumption that it's a trimer at 250, which adds up to 750. This could be asked with different levels too. So if the example I have is a protein that has a mass of 100 kilodaltons, but if you have one band at 500 kilodaltons, when ran in a gel, what size of one monomer? So one monomer would be 500 kilodaltons, and the protein would probably exist as a dimer, which is 1,000 kilodaltons. Okay. Is there any other questions you guys have?
I just want to say thank you for spending 3 hours of your evening with us, because this was super helpful. So
okay, I'm glad it was helpful. I don't know if it was too long. I think I could have condensed it down more, but it's really hard to just cover these problem sets without really going into detail on what each question is asking. So that's why it took too long. But no problem. I'm glad it helped.
Thanks, Manuel.
No problem, Angela. I'm glad it helped. All right, I'll stop the recording and upload it.